Good evening. Welcome to Rest Academy. In this session, you'll get a calm, rigorous tour of Microbiology Essentials. How can a simple purple stain divide whole kingdoms of bacteria? Why do tiny structures like capsules or spores change survival so profoundly? And how do invisible microbes living by billions sometimes turn from partners into pathogens? During this video, you'll relax while building a clear mental map of microbes and their patterns in one continuous flow. To make this journey complete, I've prepared a free practical guide with a concise summary of tonight's lesson, waiting in the description like a map you can return to whenever needed. Let's begin. You picture a night city from above, clusters of light, steady roads, quiet alleys. Microbiology is that city's atlas. It is the study of organisms too small to see unaided, and in medical settings you focus on how they coexist with you, help you, or harm you. You will meet four broad groups: bacteria, viruses, fungi, and parasites. Bacteria are single-celled prokaryotes, cells without a nucleus wrapped in a rigid wall. Viruses are acellular particles. They package genetic material but cannot replicate without a host cell. Fungi are eukaryotes, cells with nuclei appearing as yeasts, single cells, or molds, filamentous forms. Parasites are diverse eukaryotes ranging from microscopic protozoa to macroscopic helminths. In medicine, parasitology names the discipline even when some members are not strictly microscopic. You begin by separating terms that often blur in casual talk. Colonization means a microbe is present in or on you without causing damage. Infection means the microbe has entered and is replicating. Disease means that infection has led to tissue damage and symptoms you can perceive. A pathogen is a microbe capable of causing disease. An opportunist causes disease, mainly when host defenses are lowered or barriers are breached. Virulence factors are microbial traits, molecules, or structures that increase the chance of causing disease, such as a capsule preventing phagocytosis or a toxin disrupting cell signaling. A reservoir is the usual habitat where a pathogen lives and multiplies. A vector is a living carrier like a mosquito that transmits it between hosts. Hold the atlas metaphor steady. Neighborhoods are body sites. Zoning rules are physiologic conditions. Traffic laws are immune defenses. The skin is a dry acidic boundary. The gut is anaerobic and nutrient-rich. The lungs are ventilated and cleared by cilia. Microbes select neighborhoods based on what they can tolerate and what they can use. That preference is called tropism in virology, but the logic holds across groups. Receptors available, temperature, pH, oxygen, and immune surveillance decide who thrives where. You also need a naming map. For bacteria and fungi, the standard is genus and species in italics, with the genus capitalized, for example, Escherichia coli. When you do not know the exact species, you may see SPOB after the genus to indicate an unspecified member. Viral naming is different because viruses are not cells. Families and genera are capitalized but not italicized in general writing, and common names often reflect disease or morphology, for example herpes viruses. This small typographic detail prevents confusion between, say, a bacterial species and a viral illness with a similar word root. Why does this atlas matter clinically? 4 cooperative mechanisms tie the map to decisions. First, structure predicts vulnerability. A bacterial cell wall made of peptidoglycan is a target you lack, shaping classes of antibiotics conceptually tuned to walls. Second, metabolism predicts pace. A microbe's ability to use oxygen or ferment determines growth in specific sites and on specific media. Third, genetics predicts change. Horizontal gene transfer, movement of genes between microbes, alters traits like toxin production or resistance. Fourth, ecology predicts balance. Resident microbiota, your usual microbial communities, create colonization resistance by competing for nutrients and producing inhibitory molecules, restraining newcomers. Consider how host defenses weave through this. Innate immunity barriers, acidic pH, enzymes complement phagocytes responds quickly and non-specifically. Adaptive immunity, T and B lymphocytes add specificity and memory after recognition. Many symptoms you feel during infection arise from your response rather than from microbes directly. Fever is a regulated rise in the body's set point that can slow some pathogens and enhance immune efficiency. Redness and swelling are vascular adjustments, bringing cells and proteins to a site. This is why different pathogens in the same organ can produce overlapping syndromes. The organ's response creates a shared pattern, while details of timing, severity, and associated features help narrow causes. Keep the atlas practical with a short, quiet case. You wake after travel with abrupt watery diarrhea, mild cramps, and no blood. You hydrate at home and improve over a day. The pattern suggests a toxin-mediated process rather than invasive tissue destruction. Microbes that produce enterotoxins disturb ion transport in the intestine without penetrating deeply. If instead you had high fever, severe abdominal pain, or signs of dehydration, dry mouth, dizziness, minimal urine, or the stool turned bloody, that constellation would merit prompt professional care to evaluate for invasive infection or significant fluid loss. The goal here is not to diagnose you, but to model how mechanism guides attention and timing. Look closer at 2 everyday confusions you can gently resolve now. Not every microbe that enters you must be eradicated. Many are either harmless bystanders or supportive residents. In the gut, for example, resident bacteria synthesize certain vitamins and train local immunity. Disrupting them by illness or by non-selective medications can open ecological space for less friendly neighbors, a reminder that the atlas includes maintenance of borders, not only defense against invasions. Also, stronger microbes are not simply those that replicate fastest. A microbe with high virulence may burn through a host quickly, but spread poorly, while one with modest symptoms may circulate widely. Epidemiology, how infections spread, will revisit this trade-off when you calculate how many new cases arise from each case on average. A brief, composed reflection. When you name what you're seeing, colonization, infection, disease, you take restless impressions and file them neatly on the map. Naming reduces noise, mechanism reduces worry. Both invite steadier decisions. Return to the atlas and its symbols. Bacteria bring walls, ribosomes distinct from yours, and simple division. Viruses bring compact genomes, protein shells, and reliance on your machinery. Fungi bring chitinous walls and ergosterol-rich membranes. Parasites bring complex life cycles and sometimes external vectors. This overview lets you anticipate which laboratory methods will speak most clearly for each group and which therapeutics conceptually can discriminate microbes from you. You will not memorize by force. You will place each detail where it belongs on the map, so it is easy to find again. One last question hangs at the edge of the page. If structure predicts vulnerability, what single laboratory color can reframe how you approach a bacterium from the moment it grows on a plate or smears on a slide? The next section turns the page to that first vivid color choice. You start with a single slide and 4 reagents. The Gram stain, named for Hans Christian Gram, classifies bacteria by how their cell walls retain a crystal violet iodine complex. Thick peptidoglycan keeps the dye and appears purple. Thinner peptidoglycan with an outer membrane loses the dye when decolorized and takes up a red counterstain. This one-color decision, visible in minutes, divides most clinically important bacteria into gram-positive and gram-negative and instantly narrows expectations about structure, habitat, and typical mechanisms of disease. Walk through the method briefly to fix why the colors persist. First, crystal violet enters most cells. Iodine then forms a larger complex with the dye. Alcohol or acetone dehydrates thick peptidoglycan, reducing pore size and trapping the complex in gram-positive walls, while extracting lipids from the gram-negative outer membrane and increasing permeability so the complex escapes. Finally, safranin or fuchsine counterstains the now colorless gram-negatives pink to red. The outcome is not cosmetic. It predicts key features you will use later. Gram-positives often display teichoic acids embedded in their walls and lack an outer membrane. Gram-negatives possess an outer membrane with lipopolysaccharide, LPS, a periplasmic space that hosts enzymes and porins that govern entry of small hydrophilic molecules. Shape and arrangement come next, because geometry compresses possibilities. Cocci are spheres, bacilli are rods, cocobacilli are short rods with a plump outline, vibrios are curved rods, spirilla are rigid spirals, spiracetes are flexible, corkscrew-shaped. Arrangements arise from how daughter cells separate. Clusters suggest grape-like staphylococci, chains point towards streptococci, pairs diplococci evoke Neisseria or Streptococcus pneumonia, the latter often lancet-shaped in pairs. Some rods align like pickets palisades as with Corynebacterium, others branch delicately like fine twigs, Occludoactinomyces or Nocardia. With only gram color and geometry, you already begin to sort purple cauci in clusters versus purple cauci in chains, pink curved rods versus pink straight rods, slender spirals too thin to see well on a gram stain, suggesting spiracetes that need dark field or special stains. Oxygen requirements refine the map further and whisper where these organisms likely live. Oxygen generates reactive oxygen species, chemically active byproducts like superoxide and hydrogen peroxide that damage proteins, lipids, and DNA. To survive in atmospheric oxygen, microbes need enzymes that detoxify these byproducts. Superoxide dismutase, SOD, converts superoxide to hydrogen peroxide. Catalase or peroxidase then breaks hydrogen peroxide down. Obligate aerobes require oxygen and generally possess SOD and catalase. Think of environments where oxygen is plentiful: skin surface, ventilated alveoli, devices exposed to air. Obligate anaerobes cannot tolerate oxygen because they lack sufficient detoxifying enzymes. They prefer oxygen-poor niches, gingival crevices, colon, ischemic tissue, deep puncture sites. Facultative anaerobes can use oxygen for efficient respiration when available, but switch to fermentation or anaerobic respiration when oxygen is scarce. They thrive across diverse sites, including the gut and urinary tract. Microaerophiles require reduced oxygen tension compared with ambient air. They find microenvironments such as the gastric mucus layer or deep within biofilms where oxygen gradients form. Aerotolerant anaerobes do not use oxygen but possess enough detox capacity to survive brief exposure. Match these physiologies to clinical scenes you recognize. The oropharynx and colon with abundant anaerobic pockets favor anaerobes and facultative anaerobes. Foul-smelling drainage from a deep wound often reflects anaerobic metabolism, producing volatile compounds. Aspiration into the dependent lung segments can seed mixed flora, including anaerobes. In contrast, burned surfaces and indwelling catheters, bathed in air or intermittently flushed with oxygenated fluids, favor aerobes and facultative anaerobes that form robust surface communities. Understanding oxygen preferences also clarifies why abscesses, walled-off collections with poor perfusion, select for anaerobes, and why culture of anaerobes demands careful specimen handling to avoid oxygen exposure. Now add practical exceptions so the color rules do not mislead you. Some organisms stain poorly or not at all with the standard gram technique. Acid-fast bacteria such as Mycobacterium have lipid-rich walls with mycolic acids that resist decolorization by acid alcohol. They require acid-fast stains to visualize. Organisms without a classical peptidoglycan wall such as Mycoplasma and Ureaplasma do not retain gram dyes. Their membranes contain sterols and make them intrinsically different from typical gram-classified bacteria. Intracellular bacteria like chlamydia or rickettsia can be faint or invisible on routine gram stains due to small size and intracellular location, benefiting from nucleic acid amplification or specialized methods. Some bacteria are gram variable, appearing partly purple and partly pink depending on growth phase and cell wall integrity. Knowing these exceptions prevents you from overcommitting to a classification when the biology argues otherwise. Within the two big gram groups, a few quick heuristics carry weight. Gram-positive cocci. Clusters suggest Staphylococcus, chains or pairs suggest Streptococcus or Enterococcus. Gram-positive rods, club-shaped and palisading, push you toward Corynebacterium. Slender branching favors Actinomyces, anaerobic oral flora, or if weakly acid-fast, Nocardia, aerobic soil-derived. Gram-negative diplococci. Kidney bean pairs suggest Neisseria. Encapsulated, coffee bean pairs in the respiratory tract may signal Moraxella catarrhalis. Gram-negative rods, lactose fermentation on differential media sorts Escherichia and other enterics from non-fermenters like Pseudomonas, but that laboratory layer belongs to diagnostics. At the bedside, the immediate cues are shape, straight versus curved, and habitat, gut versus water devices, with curved motile oxidase-positive rods pointing toward Vibrio or Campylobacter. Spirochetes, Treponema, Borrelia, Leptospira hide from gram stains yet declare themselves through motion patterns and serologic footprints. Return to oxygen one more time because it synthesizes shape, color, and place. Obligate anaerobes flourish where circulation is poor and tissues are devitalized. They often participate in polymicrobial infections with synergistic partners that lower the redox potential. Facultative anaerobes can carve the initial path, consuming oxygen and paving the way for stricter anaerobes. Aerobes exploit surfaces with steady oxygen and often possess pigments or oxidative enzymes that generate colorful colonies and oxidative stress resistance. When you read a report noting mixed anaerobic flora from a deep dental space, the physiology explains both the smell and the need for drainage, rather than relying on oxygen-sensitive agents alone. When you see non-fermenting gram-negative rod from a catheter, the aerobic habitat and biofilm tendency align with the device story, without naming a drug. A brief cameo from the clinic keeps the map real. After a molar extraction, a patient develops progressive jaw pain, swelling, trismus, and a thick malodorous exudate. The anatomic space is deep, perfusion is limited, and oral anaerobes predominate. Worsening fever, difficulty swallowing, or breathing concerns would be cues to seek urgent professional care for assessment of deep space infection and airway risk. The value for you here is not to guess names, but to correctly predict this is likely anaerobic and mixed, guiding specimen handling and early precautions while definitive tests proceed. A short reflection to settle the concept: 2 colors, a silhouette, and a breath test. Gram, shape, and oxygen turn a shapeless crowd into a legible cast. With those 3, you hear the case differently and place it on the right street of the atlas. You close this section by noticing what your classification implies about parts you have not yet examined. Gram-positive walls that trap dye, gram-negative outer membranes that host LPS, shapes that suggest motility or pairing, oxygen habits that demand or avoid specific enzymes, all point downstream to physical structures, peptidoglycan thickness, capsules that blur outlines, flagella that drive motion, and spores that survive harsh hours. The next page opens those doors and lets you step inside the cell's architecture to see how each structural piece earns its place on the map. You study a compact fortress under high magnification. The outer wall, the gates, the pennants, the sealed vault, each part is small, specific, and purposeful. Bacterial structure is the architecture that predicts both vulnerability and behavior. Begin with the wall. Peptidoglycan is a rigid mesh unique to bacteria, built from repeating disaccharides N-acetylglucosamine and N-acetylmuramic acid, cross-linked by short peptides. Enzymes called penicillin-binding proteins catalyze the crosslinks. Their activity sets how strong the lattice becomes. In gram-positive bacteria, the peptidoglycan layer is thick and lies at the surface. In gram-negative bacteria, a thinner layer sits between 2 membranes. Fix the layers in one glance. Gram-positives display teichoic acids, polymers anchored in the wall, and lipoteichoic acids, variants anchored in the membrane. These acids help maintain wall structure, influence capsion binding, and can stimulate host inflammation upon release. Gram-negatives add an outer membrane rich in lipopolysaccharide, LPS. LPS has 3 parts: O-antigen, a repeating polysaccharide that varies among strains, a core oligosaccharide, and lipid A, the membrane anchoring moiety that when detected by the host can provoke strong inflammatory responses. Porins, protein channels in the outer membrane, admit small hydrophilic molecules. Their selectivity shapes what can enter. A thin periplasmic space between outer and inner membranes hosts enzymes that modify nutrients and clinically relevant may inactivate antibiotics. Every fortress needs a boundary within. The cytoplasmic membrane is a phospholipid bilayer studded with proteins that manage transport, sense conditions, and house energy systems. Bacteria lack mitochondria. Instead, energy-transducing electron transport chains reside in this membrane, establishing proton gradients that drive ATP synthesis. Because these processes sit at the membrane, damage to membrane integrity can stall core metabolism quickly. Some bacteria sheath themselves in an additional cloak. A capsule is an organized, tightly attached layer, most often polysaccharide, sometimes polypeptide, that protects against desiccation and shields from immune recognition and ingestion. The classic protein exception is the poly-D-glutamate capsule of Bacillus anthoracensis. A related but looser coating, the slime layer, forms part of the glycocalyx. It is more easily washed away, yet crucial for surface attachment and early biofilm steps. Capsules can be visualized indirectly by reactions that make them swell, a quelling effect, or by negative contrast methods. In the clinical laboratory, antigen detection from encapsulated organisms can also signal their presence. Functionally, capsules impede opsonization, the tagging of bacteria for phagocytosis, by masking surface structures and interfering with complement deposition. Fortresses communicate and grapple. Pilia, also called fimbria, are thin, hair-like protein projections that mediate adherence to host tissues and abiotic surfaces. Adhesins, the specific binding proteins at tips or along the shaft, recognize receptors such as glycoproteins on mucosal cells. This host microbe fit helps explain tissue tropism. A specialized longer pilus, the sex pilus, connects donor and recipient bacteria during conjugation to transfer DNA. You will return to that exchange when you explore genetics. Gram-negative pathogens also assemble secretion systems, multi-protein conduits spanning membranes that deliver effector proteins into host cells or neighboring bacteria. One well-known example, the type 3 secretion system, operates as a molecular syringe that alters host cell signaling to favor colonization or invasion. Other systems, types 4 and ZIs, move proteins or DNA across boundaries, shaping competition and virulence. Movement changes how a fortress explores terrain. Flagella are helical filaments turned by rotary motors embedded in the membrane and wall. They generate propulsion through liquid and allow chemotaxis, movement along chemical gradients. Arrangements differ. Monotrichous, single polar, Lophotrichous, tufts at a pole, Amphitrichous, one at each pole, or Paratrichous, distributed. The pattern can hint at identity and niche. Spirochetes solve movement differently. Their flagella reside within the periplasm as axial filaments that wrap around the cell cylinder. When they rotate, the entire organism corkscrews, allowing travel through viscous media like mucus or connective tissue. Inside the fortress, compact efficiency rules. The nucleoid is the region where the chromosome, typically a single circular DNA molecule, resides without a surrounding membrane. Ribosomes, the protein synthesis machines, are 70S particles made of 50S and 30S subunits. Their RNA and protein compositions differ enough from human 80S ribosomes to allow selective targeting by certain antibiotic classes. Plasmids are small, extrachromosomal DNA circles that can carry traits affecting metabolism, virulence, or resistance. Copy number and mobility vary by plasmid type. Inclusion bodies, storage granules for carbon, phosphate, or sulfur, reflect environmental history and upcoming needs. Some fortresses endure siege by becoming seeds. Endospores are dormant, highly resistant structures formed by certain gram-positive genera, notably Bacillus and Clostridium. An endospore contains a dehydrated core with DNA, small acid-soluble proteins that stabilize the genome, high levels of calcium and dipecalinic acid that further protect and compress the core, a thick cortex of modified peptidoglycan, and a multi-layered coat rich in keratin-like proteins. Endospores do not metabolize and are not reproductive. A single vegetative cell yields one endospore, which germinates back into one vegetative cell when conditions improve. Their resistance to heat, desiccation, chemicals, and radiation explains persistent environmental survival and the need for high-level sterilization to eliminate them. Special stains, such as malachite green with heat, highlight spores as green ovals inside pink counterstained cells or free in the field. Not all walls behave the same under dyes. Acid-fast bacteria, exemplified by Mycobacterium, build a wall with peptidoglycan linked to arabinogalactan and overlaid with mycolic acids, long-chain fatty acids that create a waxy barrier. This structure resists decolorization by acid alcohol and many common disinfectants, explaining the need for acid-fast stains and particular laboratory handling. Some bacteria assemble an S-layer, a crystalline protein lattice external to the wall that can contribute to surface resilience and immune evasion. Others, such as Mycoplasma, forego a cell wall entirely and stabilize their membranes with sterols acquired from the host, a structural choice that alters susceptibility patterns and staining. Structure predicts encounter with host defenses. Teichoic acids and fragments of peptidoglycan are recognized by innate immune receptors, and can trigger cytokine release. LPS lipid A binds specific host sensors and can activate complement and coagulation pathways. Capsules conceal these signals, reduce complement deposition, and can block binding to phagocyte receptors. Porins limit entry of large or hydrophobic compounds. Changes in porin expression can reduce susceptibility to agents that normally pass through them. Periplasmic beta-lactamases, enzymes that cleave beta-lactam rings, sit precisely where incoming molecules are most vulnerable. Secretion systems deliver proteins that rearrange host cytoskeleton, disrupt tight junctions, or inhibit phagosome maturation, all structural acts with functional consequences. A brief clinical scene ties pieces together. A middle-aged patient without a spleen develops sudden fever, rigors, and confusion hours after a minor dog bite. Think architecture. An encapsulated organism with potent antiphagocytic armor gains entry from the oropharynx of the animal. Absent splenic filtration reduces clearance of encapsulated cells. Rapid progression, altered mental status, or rash would be cues to seek immediate professional care to evaluate for fulminant sepsis. The take-home for you is the structural logic. Capsule-defended invaders exploit a missing checkpoint. A quiet reflection, then back to the walls. When you look past names and see layers, gates, and flags, the chaos settles. You are not memorizing lists. You are reading blueprints, and blueprints do not change overnight. You close by connecting architecture to tempo. Thick walls, protective coats, efficient gates, motile drives, and dormant seeds explain who endures dryness, who rides fluids, and who waits out hardship. But structure alone does not fill granaries or fuel expansion. What happens when nutrients arrive at the gate? Sensors flip, and the internal engines choose between rapid division and slow persistence. The next section follows the fortress into its growth phases and metabolic strategies, where time and resources set the rhythm. You watch a curve grow like a quiet season on paper, a flat line, a rising hill, a long plateau, then a gentle descent. That curve, population over time, captures how bacteria adjust to new conditions. The first stretch is lag phase, when cells sense their surroundings, repair damage, and synthesize enzymes for the nutrients at hand. Numbers barely change, but machinery resets. Exponential phase follows. Cells divide at a characteristic generation time, the steepest slope on the curve. Stationary phase arrives when nutrients limit or waste accumulates, growth balances death, and metabolism shifts to maintenance. Death phase closes the cycle as viable counts fall under stress. You are not memorizing labels, you are reading the tempo of a living system. Hold the farm image steady. In lag phase, bacteria survey the soil, carbon sources, nitrogen, minerals, and tune gene expression before planting. In exponential phase, harvest is brisk, glycolysis and related pathways run fast, precursors feed biosynthesis, and cell walls expand at their growth zones. Stationary phase is drought management. Stress responses rise, membranes stiffen, and scavenging systems turn on. In death, spores, if a species can form them, wait like hardy seeds for rain. The same field yields different crops depending on what fertilizers and water you deliver, and the same medium nurtures different microbes depending on what they can digest and how they balance energy. Define the inputs with care. Carbon is the backbone of cell material. Nitrogen builds amino acids and nucleotides. Phosphorus and sulfur support nucleic acids and certain amino acids. Trace metals serve as enzyme cofactors. Many bacteria secrete cytophores, high-affinity iron-capturing molecules, because free iron is scarce in hosts. Water activity. The effective availability of water constrains life. High salt or sugar lowers it, so halotolerant microbes endure where others stall. Temperature preferences sort species. Human-associated mesophiles prefer the moderate range of your tissues. Environmental specialists may favor colder or hotter niches. pH shapes enzyme stability. Acid-adapted microbes thrive in low pH pockets, whereas alkalophiles exploit high pH environments. Oxygen and redox potential, capacity to accept electrons, decide whether respiration or fermentation pays best in the moment. Name energy choices clearly. Respiration couples the oxidation of electron donors, often reduced carbon compounds, to the reduction of electron acceptors via an electron transport chain in the cytoplasmic membrane. This chain pumps protons outward and stores energy as a proton motive force, which returns through ATP synthase to make adenosine triphosphate, ATP, the cell's energy currency. Aerobic respiration uses oxygen as the terminal electron acceptor. Anaerobic respiration uses alternatives such as nitrate or sulfate. Fermentation is different. It generates ATP at the level of substrate breakdown, then reoxidizes reduced cofactors like nicotinamide adenine dinucleotide (NADH) by producing organic end products, acids, alcohols, or gases without an external electron acceptor. Respiration usually yields more ATP per carbon than fermentation, but fermentation buys independence from external acceptors and supports rapid bursts when oxygen is scarce. Zoom inside the pathways only as far as needed to read the season. Glycolysis converts glucose to pyruvate with modest ATP gain and reduced cofactors. Variants like the Entner-Jodorov route appear in some gram-negative bacteria. When respiration is possible, pyruvate or its derivatives flow into the tricarboxylic acid TCA cycle to generate more reduced cofactors, and the electron transport chain harvests their energy. When respiration is blocked or acceptors run out, pyruvate becomes end products that regenerate oxidized cofactors, keeping glycolysis turning. In stationary phase, bacteria often pull carbon from storage polymers, shift to stress-protective metabolites, and deploy detox systems for reactive oxygen species if oxygen lingers. Return to the field plan and consider growth media conceptually. Minimal media supply defined amounts of essentials. Only bacteria with the right biosynthetic tools can flourish. Rich or complex media provide amino acids, vitamins, and growth factors. Fastidious organisms, those with many nutritional requirements, grow here while they falter on bare-bones recipes. Selective media include inhibitors to suppress some groups and favor others. Differential media reveal metabolic traits by color changes. You do not need brand names to use the logic. Composition filters the cast, and readouts translate metabolism into visible clues. Tie metabolism to habitat like rainfall to harvest. In the gut, low oxygen and abundant fermentable substrates favor organisms that excel at fermentation and at tolerating organic acids. In the alveoli, oxygen and surfactant shape a different guild. Organisms with robust oxidative defenses and surface attachment skills compete better. Ischemic tissue, poorly perfused, low in oxygen, selects for anaerobes and creates a chemical environment that blunts some host defenses. Indwelling devices add another niche. Smooth materials, intermittent flow, and conditioned surfaces invite adherent communities rather than free-swimming cells. Those communities are biofilms, structured, surface-attached populations encased in a self-produced matrix. The matrix, often called extracellular polymeric substance, is a blend of polysaccharides, proteins, and extracellular DNA that anchors cells and creates microenvironments. Biofilm development typically moves through stages: initial reversible adhesion, irreversible attachment with matrix production, microcolony formation, maturation into three-dimensional structures with water channels, and dispersal of cells to seed new sites. Within the matrix, gradients form: oxygen high at the surface, low deep inside, nutrients plentiful at channels, scarce in pockets. Cells in different layers adopt different metabolisms: fast growers at the edge, slow or dormant cells deeper, including persister cells, metabolically quiet variants that survive stress without inheriting classical resistance genes. Why do biofilms matter to you? 3 mechanisms carry outsized clinical consequences. First, limited penetration. The matrix and its chemistry slow the diffusion of many agents, so surface layers see higher concentrations than the core. Second, altered physiology. Slow growth and stress programs reduce the efficacy of agents that target active division or specific pathways. Third, collective defense. Enzymes in the periphery can inactivate compounds before they reach the interior, and coordinated signaling quorum sensing can adjust behavior across the community. The outcome is tolerance, reduced killing under exposure, distinct from inherited resistance. When exposure ends, survivors can repopulate, sustaining chronic, relapsing patterns. Anchor these ideas with a calm case. A person with a long-term urinary catheter notices cloudy urine and intermittent low fevers over weeks. Cultures repeatedly grow the same organism despite changes in technique. The story fits a surface-attached community in a device lumen. Intermittent flow and nutrient pulses favor matrix-embedded cells that shed into the stream. New flank pain, high fever with chills, vomiting or confusion would be signals to seek prompt professional care to evaluate for ascending infection or systemic involvement. The map you carry forward is ecological. Surface plus flow plus time equals biofilm risk. Metabolism also predicts the gases you smell and the acids that sting. Anaerobic fermentation can generate volatile compounds that carry a characteristic odor. Oxidative pathways may produce pigments that mark colonies. Carbon sources shape outputs. Some species switch pathways when given alternate sugars, a strategy called catabolite repression that prioritizes the most efficient fuel first. Stress responses in stationary phase strengthen cross-protection, so prior exposure to one mild stress can incidentally prepare cells to face another, a phenomenon known as adaptive resistance at the physiological level, again distinct from stable genetic change. A brief reflection to settle the scene. When you watch the seasons rather than snapshots, confusion quiets, growth phase, energy choice, and community shape explain tempo, smell, texture, and persistence, practical signals that turn reports into stories you can follow. Close the gate on this field with one last thought about surprise harvests. Overnight, a colony begins to use a sugar it could not use yesterday, or a once vulnerable population endures an exposure it once could not bear. What changed is not the weather, it is the instructions. Next, you watch those instructions move, by uptake, by phage, by bridge, and see how genes redraw tomorrow's map. You picture a quiet library, where volumes slip from one shelf to another, sometimes by invitation, sometimes by theft, sometimes by delivery in sealed packages. Bacterial genetics is this exchange, and its fluidity explains sudden shifts in traits, new toxins, new resistances, new metabolic tricks appearing within familiar organisms. Unlike eukaryotes, bacteria reproduce mainly by binary fission, one cell dividing into two genetically identical daughters. That vertical transmission sustains the lineage. Yet bacteria also excel at horizontal gene transfer, moving DNA sideways across lineages. The three major routes are transformation, transduction, and conjugation. Each has distinct mechanisms but converges on the same outcome, an altered genetic repertoire that may confer survival advantages. Begin with transformation, the simplest library exchange. Some bacteria become competent, able to take up free DNA fragments from the environment. Competence may be natural, regulated by quorum sensing and stress signals, or induced under laboratory conditions. DNA released from lysed cells floats like loose pages. A competent cell binds and internalizes it, sometimes integrating it into its chromosome by recombination if sequences align. In clinical context, think of Streptococcus pneumonia acquiring capsule genes, producing new serotypes that evade immunity and vaccines. Transformation requires no live donor, only available DNA and a receptive recipient. Next is transduction, in which bacteriophages, viruses that infect bacteria, act as couriers. Two flavors matter. Generalized transduction occurs when a phage accidentally packages host DNA fragments during assembly and delivers them into the next bacterium it infects. Specialized transduction occurs when a temperate phage, integrated into the bacterial chromosome as a prophage, excises imprecisely, carrying adjacent host genes with it into new infections. This mechanism has transferred toxin genes historically. The diphtheria toxin gene resides on a phage infecting Corynebacterium diphtheria. Cholera toxin genes lie within lysogenic phages infecting Vibrio cholerae. Without the phage, those bacteria would not cause classic disease. With the phage, virulence emerges. Conjugation resembles a direct bridge between cells. A donor with a fertility plasmid, F plasmid, extends a sex pilus, attaches to a recipient, and transfers DNA through a type OIF secretion system. Sometimes only the plasmid itself moves, sometimes a conjugative plasmid integrates into the chromosome and transfers adjacent chromosomal segments. This horizontal sharing can cross species boundaries, allowing resistance determinants to disseminate across genera. Conjugative plasmids often carry multiple resistance genes in clusters, turning a once sensitive organism into a multi-drug resistant strain after a single exchange. Hospitals see the practical outcome in enteric bacteria swapping, extended spectrum beta-lactamase genes or carbapenemase genes, spreading them rapidly. Plasmids deserve special focus. These small, usually circular DNA molecules replicate independently of the chromosome. Some plasmids are non-conjugative, but can hitchhike with conjugative plasmids. They often encode accessory traits, resistance, virulence factors, or metabolic capacities, not essential for growth under standard conditions, but critical in stress or host environments. Copy number varies. High copy plasmids yield abundant gene products quickly. Low copy plasmids conserve resources, but may be more stably maintained. Plasmid stability systems, like toxin-antitoxin pairs, ensure persistence within populations by killing cells that lose them. This coercive mechanism means a plasmid can become inseparable from its host's future. Genetic rearrangements also occur internally. Transposons, mobile DNA segments, jump between chromosomal and plasmid sites, carrying resistance or virulence genes with them. Insertion sequences are the simplest, encoding only the transposose enzyme. Composite transposons bracket additional genes between insertion sequences. Integrons collect gene cassettes, small mobile elements into arrays, particularly resistance genes, and can be mobilized by transposons or plasmids. The genetic architecture resembles modular furniture. Pieces fit together in new configurations, rapidly altering phenotype without changing the core genome. Mutation, while less dramatic than transfer, remains steady. Background noise, errors during DNA replication, or damage from reactive chemicals and radiation generate changes. In bacteria, haploidy means every mutation is expressed, no second allele to mask it. Mutations that reduce binding sites for antimicrobials or modify metabolic enzymes can yield resistance or alter virulence. Combined with selection pressure from environment, host immunity, or antimicrobial use, these mutations either vanish or sweep through populations. Horizontal transfer then redistributes successful innovations widely. Tie the library back to patient care with a focused example. A young adult presents with sore throat, fever, and a gray pseudomembrane in the throat, classically diphtheria. The responsible bacterium, Corynebacterium diphtheria, is not inherently toxic. Only strains lysogenized by a phage carrying the toxin gene cause severe disease. Here, virulence is borrowed text, inserted by transduction. A subtle shift, phage presence transforms a benign colonizer into a lethal invader. For you, the clinical lesson is that genes may not align with species boundaries. Context, phage, plasmid, or transposon writes the script. New neurologic symptoms, airway compromise, or myocarditis in such a scenario would be red flags requiring immediate professional evaluation. Reflect gently. The idea that instructions move and recombine explains why yesterday's patterns may not hold tomorrow. Instead of confusion, you gain an explanation. Traits are modular, passed hand to hand in microbial society. Close this section with the anticipation that borrowed genes become weapons or cloaks. Once acquired, how do microbes deploy these instructions to breach barriers, attach to cells, evade defenses, or produce toxins? The next chapter walks through pathogenicity, the strategies microbes use to convert genetic instructions into actual disease. You step into a dim chamber where tools hang on hooks, grappling lines, ladders, acid vials, smoke bombs. These are not human weapons, but microbial strategies, pathogenicity factors that turn colonization into harm. Pathogenicity is the ability to cause disease. Virulence is the degree of that ability. The factors are not random. Each addresses a barrier you place in the microbe's path. Adhesion to resist washing away, invasion to breach surfaces, evasion to escape immunity, toxins to disrupt cellular function. Start with adhesion, the grappling line. To remain at a mucosal surface, a bacterium must resist physical clearance by fluid flow, ciliary action, or peristalsis. Fimbria and pili are thin protein filaments tipped with adhesins, proteins recognizing specific receptors on host cells. The specificity explains tissue tropism. Uropathogenic Escherichia coli express P. fimbria that bind receptors on uroepithelial cells, favoring the urinary tract. Adhesins may also reside in surface proteins or lipoteichoic acids in gram-positive bacteria, providing direct binding to extracellular matrix proteins such as fibronectin or laminin. Once bound, bacteria can resist being flushed and begin to form communities. Next are invasins, the ladders and picks. These molecules enable bacteria to penetrate host cells or spread through tissues. Some invasins manipulate host cytoskeleton to induce endocytosis. Yersinia species use invasins to trigger uptake by non-phagocytic cells. Listeria monocytogenes expresses internals that bind host cadherins, stimulating entry. Once inside, bacteria may escape the endocytic vacuole into cytoplasm, as Listeria and Shigella do, allowing actin-based motility and spread to adjacent cells without extracellular exposure. Other invasins degrade extracellular matrix, such as hyaluronidase, breaking down connective tissue barriers, easing spread through tissues. Capsules act as cloaks, masking bacterial surfaces. Polysaccharide capsules prevent opsonization and phagocytosis, hiding antigens from recognition. Encapsulated pathogens, Streptococcus pneumoniae, Haemophilus influenzae type B, Neisseria meningitis cause invasive diseases such as meningitis and sepsis, especially in hosts with impaired splenic clearance. Some capsules mimic host molecules. Neisseria meningitis serogroup B capsule resembles neural cell adhesion molecules, reducing immune recognition. Beyond capsules, bacteria produce proteins that bind host immunoglobulins in a non-functional orientation. Staphylococcus aureus protein A binds the Fc region of IgG, preventing opsonization. Streptococcus pyogenes M protein interferes with complement deposition. Now the smoke bombs, toxins. Exotoxins are proteins actively secreted by bacteria. They are often highly potent and specific. They may be classified by action. AB toxins consist of an active A subunit and a binding B subunit. Diphtheria toxin inactivates elongation factor 2, halting protein synthesis. Cholera toxin activates adenylate cyclase through G protein modification, raising cyclic AMP and causing fluid secretion. Membrane-damaging toxins form pores, disrupting cell membranes. Streptolis and O perforates red and white blood cells. Superantigens such as Staphylococcus aureus toxic shock syndrome toxin cross-link T-cell receptors to MHC molecules indiscriminately, triggering massive cytokine release and systemic shock. Endotoxin, in contrast, is not secreted but embedded in gram-negative outer membranes, the lipid A component of lipopolysaccharide, LPS. When gram-negative bacteria lyse, lipid A is released and recognized by host toll-like receptors. In low amounts, it triggers local inflammation. In high amounts, it provokes systemic effects, fever, hypotension, disseminated intravascular coagulation, collectively called endotoxic shock. The difference between exotoxin and endotoxin is functional. Exotoxins are proteins with specific cellular targets. Endotoxin is a lipid trigger of host inflammatory cascades. Secretion systems sharpen delivery. Gram-negative bacteria deploy syringe-like complexes. The type 3 secretion system injects effectors into host cells, altering cytoskeleton and immune signaling. Salmonella and Shigella rely on this to invade intestinal epithelium. Type 4 systems can transfer DNA or proteins used by Helicobacter pylori to inject CAGA protein into gastric epithelial cells, promoting inflammation and oncogenic change. These systems make delivery precise, bypassing the extracellular milieu. Quorum sensing underlies coordination. Bacteria release small signaling molecules that accumulate as population density rises. When a threshold is reached, gene expression shifts collectively. Virulence factors switch on, biofilm programs activate, toxins release in synchrony. Pseudomonas aeruginosa uses quorum sensing to regulate elastase and piocyanin production, timing damage to moments when its population can overwhelm host defenses. The logic is communal: act only when numbers ensure impact. Tie the weapons to host response. Many symptoms come not from direct bacterial action, but from your immune reaction to these factors. Fever, hypotension, leukocytosis, or rash can signal systemic immune activation to bacterial components. Local swelling and pain often reflect immune cells responding to bacterial enzymes and toxins. The balance between microbial strategy and host defense determines whether colonization remains quiet or escalates into overt disease. A calm clinical cameo helps anchor this. A previously healthy adult develops sudden high fever, stiff neck, and altered consciousness. The likely culprits are encapsulated bacteria breaching the bloodstream and meninges, Neisseria meningitis, Streptococcus pneumoniae. The capsule allows escape from clearance, while inflammatory responses to their wall and toxins provoke swelling and vascular compromise. Headache with photophobia and confusion in this context would be reasons for immediate professional evaluation. For you, the focus is the structural logic. Capsule plus toxins plus host inflammation explains the rapid syndrome. A short reflection closes the armory. When you see disease not as chaos but as tools applied in sequence, adhesion, invasion, evasion, intoxication, the story untangles. Symptoms are not random, they are footprints of strategies. You leave this chamber of weapons with one more question. If bacteria wield grappling lines, cloaks, and toxins, what about entities that are not cells at all? Viruses lack walls, ribosomes, and metabolism, yet they commandeer yours. Next, you turn to virology and classify these subcellular agents by the very material they carry and the coats they wear. You hold a tray of tiny parcels and read their labels in a steady light. Virology classification is simply a disciplined way to sort these parcels before you watch what they do. Two labels matter first: the kind of genetic material inside, and whether a lipid envelope surrounds the shell. From those labels flow stability, transmission, favored entry routes, and the kinds of cells a virus can exploit, its tropism. Open the tray with the genome tag. A genome is the full set of genetic instructions. Viruses package either DNA or RNA, not both, and the strand can be single or double. You can also ask whether the RNA reads directly as messenger RNA or must be transcribed before translation. This logic is organized formally in the Baltimore scheme, which groups viruses by genome type and replication strategy. Double-stranded DNA, dsDNA. Viruses generally use host nuclear polymerases. Single-stranded DNA, ssDNA. Viruses must make a complementary strand first. Double-stranded RNA, dsRNA. Viruses carry or encode an RNA-dependent RNA polymerase because your cells do not provide one. Positive sense single-stranded RNA, COC and ps-sRNA genomes function like mRNA on entry. Negative sense single-stranded RNA, COC and ps-sRNA genomes are the opposite polarity and must be transcribed into positive sense RNA by a viral polymerase delivered in the particle. Two hybrid categories use reverse transcription. Retroviruses carry pol-ssRNA that is copied into DNA by reverse transcriptase, and hepatinoviruses carry gap dsDNA that is repaired in the nucleus and transcribed through an RNA intermediate before reverse transcription back to DNA. With only this map, what the genome is and what enzymes it needs, you can already anticipate where replication will occur and which steps are non-negotiable. Now check the outer packaging. Every variant has a capsid, a protein shell built from repeating units, capsimers, arranged with geometric economy. Icosahedral capsids enclose genomes in near-spherical symmetry. Helical capsids wrap genomes in flexible coils. A few families build complex shapes. Many animal viruses add a lipid envelope outside the capsid, derived from host membranes during budding and studded with viral glycoproteins that mediate attachment and entry. Envelope status divides behavior cleanly. Enveloped viruses are fragile in the environment. Lipids dissolve with detergents, heat, and desiccation, and thus tend to spread by close contact, respiratory droplets, or body fluids. Non-enveloped viruses are tougher. Protein shells resist drying and surfactants, and thus survive on hands, fomites, and through the gastrointestinal tract where bile salts and acid would disrupt a lipid envelope. When you hear about outbreaks traced to cruise ships or daycare surfaces, you are often dealing with non-enveloped RNA viruses that tolerate the journey. When you consider viruses requiring intimate contact, an envelope is often part of the story. Add two structural corollaries you will use later. In human pathogens, helical nucleocapsids are almost always found inside an envelope. Naked helical particles are common in plants but not in you. And envelope glycoproteins are the keys for cell entry. Antigenic variation in these spikes can alter receptor binding and immune recognition, reshaping epidemiology without changing the internal replication machinery. Tropism is next. Which hosts, which tissues, which cells? Tropism means preference based on compatibility at several levels. At the surface, viral attachment proteins must bind a specific receptor, often a protein or a glycan, on the host cell. If the receptor is absent, the virus cannot attach. If it is present only on certain cells, those cells become preferred targets. Beneath attachment, the interior must support the virus's entry route, uncoding, genome expression, replication, and assembly. Nuclear localization signals may be needed for DNA viruses that use host enzymes. Cytoplasmic factories must be available for RNA viruses that replicate outside the nucleus. Cellular proteases may be required to activate fusion proteins. Innate antiviral programs may be weak or strong in a given tissue. The net effect is selective. A respiratory epithelium with the right receptors and proteases will be a receptive landing strip. A neuron with limited turnover and specific transport pathways may be a long-term refuge. Place replication in the cell without stepping through the whole cycle yet. DNA viruses generally replicate in the nucleus because they exploit host DNA polymerases and transcription machinery. Poxviruses are the major exception, carrying a full toolkit for cytoplasmic replication. RNA viruses generally replicate in the cytoplasm using viral RNA-dependent RNA polymerases. Orthomyxoviruses are a key exception, importing their segmented RNA genomes into the nucleus to exploit host-capped RNA fragments for transcription. Retroviruses reverse transcribe their RNA into DNA, integrate into the host genome, and then use host machinery for transcription. That integration creates options for latency, where the viral genome persists silently between episodes of production. Genome architecture adds another layer. Segmented genomes, distinct RNA pieces packaged in one virion, enable reassortment when two related viruses co-infect a cell, mixing segments into new constellations in a single step. This mechanism produces sudden antigenic jumps that can redraw population susceptibility. Non-segmented genomes cannot reassort, but still drift through point mutations over time, especially in RNA viruses whose polymerases lack proofreading. The mutation rate and selection pressures host immunity, receptor availability, transmission pathways, shape antigenic change, altering how often you meet a new enough version to become susceptible again. Combine these features into recognizable clinical patterns. Non-enveloped, positive-sense RNA viruses that resist acid and bile often favor fecal-oral transmission and cause gastroenteritis. Their stability explains explosive spread in confined settings and resilience on surfaces. Enveloped respiratory viruses, by contrast, spread efficiently in droplets and aerosols but fall off surfaces quickly. Their environmental fragility concentrates spread in close contact, indoor air, and seasonally favorable humidity. Neurotropic viruses display tropism for neurons or supporting cells, sometimes entering at peripheral sites and moving along axons to the central nervous system. Their capsids and entry proteins often exploit neuronal receptors or transport machinery. Hepatropic viruses target hepatocytes, reflecting receptor presence and a replication strategy that fits the liver's biosynthetic environment. A brief, focused cameo makes this concrete. In midwinter, a household develops abrupt vomiting and watery diarrhea within a day of sharing a buffet. The short incubation, fecal-oral spread, and hardiness on surfaces point toward a non-enveloped RNA virus tolerant of acid and detergents. If instead the story centered on painful grouped vesicles following close contact and reactivation during stress, you would think of an enveloped DNA virus that establishes latency in sensory ganglia and requires skin-to-skin transmission. In either scenario, persistent high fever, signs of dehydration, dizziness, decreased urine, confusion, or severe abdominal pain would be reasons to seek prompt professional care for assessment and support. The classification you performed silently, envelope, genome, tropism, guided your expectations before any test returned. Hold one gentle reflection. When you name a parcel's material and wrapping, the rest of the behavior follows. Stability, route, and preferred doorways are not mysteries, they are consequences of structure. You finish the tray with a glance at the labels you have learned to read. DNA or RNA, single or double, positive or negative sense, segmented or not, enveloped or naked, helical or icosahedral. Each label constrains what can happen next, and those constraints make the replication story manageable. The next step is to watch one parcel from handshake to departure, attachment, entry, uncoding, genome expression, assembly, release, and to see how a virus chooses between brief, lytic bursts and long, quiet latency that waits in your cells like a letter never mailed. What precisely turns that letter over and starts the press? You follow a small parcel to a loading dock. then into a humming print room where rollers, dyes, and binders convert a single template into thousands of copies. Viral replication is this factory: attachment, entry, uncoding, expression, genome replication, assembly, and release, driven by the rules of the parcel's genome and the presence or absence of a lipid envelope. Attachment is the handshake. A viral attachment protein on the capsid or envelope binds a specific receptor on the host cell. Sometimes a co-receptor is required to stabilize entry. Receptors are ordinary cell molecules, proteins or glycans with day jobs in signaling or transport. Binding determines host range and tissue tropism, and avidity matters. Multiple weak interactions can add up to a strong hold when many spikes engage the surface. Entry is the doorway. Enveloped viruses can fuse their membrane with the plasma membrane at the surface, or fuse from within an endosome after receptor-mediated endocytosis and acidification. Fusion proteins undergo pH or receptor-triggered conformational changes that bring membranes together. Non-enveloped viruses generally enter by endocytosis and then perforate or disrupt the endosomal membrane using capsid changes or lytic peptides to reach the cytosol. Some capsids form channels to thread the genome directly across a membrane. Others disassemble stepwise to reveal internal peptides that punch a way out. Uncoding releases the genome, initiating the eclipse period, the interval when infectious particles are undetectable because the incoming variants have disassembled, but new ones have not yet formed. Where uncoding occurs depends on the virus. DNA viruses that rely on host polymerases often transport their genomes into the nucleus. Many RNA viruses uncode in the cytosol to keep their replication machinery close to ribosomes and membranes. Expression and genome replication follow genome logic. Double-stranded DNA viruses typically use host RNA polymerase II to transcribe early genes in the nucleus, then replicate DNA using host or viral DNA polymerases. Large DNA viruses encode their own polymerases and replication factors. Poxviruses go further, carrying a full toolkit that allows entirely cytoplasmic replication within virus-built factories. Single-stranded DNA viruses must first synthesize a complementary strand to form a double-stranded template before transcription and replication proceed. Positive-sense single-stranded RNA, GAT-imposter RNA genomes act as ready mRNA on entry. Ribosomes translate them immediately. Many poser RNA viruses produce a large polyprotein that is cleaved by viral proteases into functional components. Others generate subgenomic RNAs to control protein ratios. Replication requires a viral RNA-dependent RNA polymerase that copies the poser RNA into a negative template and then back into new poser RNA genomes. Negative-sense single-stranded RNA, Bacterial RNA genomes are the mirror image of mRNA and must be transcribed into positive sense by a viral polymerase packaged inside the incoming virion. Without that polymerase, no proteins can be made. Segmented RNA viruses often replicate in the cytoplasm, but orthomyxoviruses carry their segments into the nucleus to snatch steel-capped RNA fragments from host transcripts to prime synthesis of viral mRNAs. Double-stranded RNA viruses keep their genomes inside a partially intact capsid, while their RNA-dependent RNA polymerase transcribes positive strands. Exposing long dsRNA in the cytosol would trigger potent antiviral alarms. Retroviruses package PLOS RNA but first reverse transcribe it into DNA via reverse transcriptase. The resulting DNA integrates into the host genome as a provirus, which host enzymes then transcribe into mRNA and new genomes. Epidemiviruses carry gapped dsDNA, repair it in the nucleus to a stable circular form, transcribe RNA intermediates, and reverse transcribe those back into DNA during assembly. Genome replication is chained to error rates and segment logistics. RNA-dependent RNA polymerases lack proofreading in most families, generating frequent mutations that create a cloud of related variants. Selection filters this swarm. Segmented genomes must package the full set of segments. Assembly signals on each segment coordinate inclusion, and when two strains co-infect the same cell, segments can reassort to form new combinations in one step. Assembly organizes parts at specific sites. Capsid proteins self-assemble with remarkable fidelity, guided by scaffolds or packaging signals on genomes. Enveloped viruses embed their glycoproteins in a host membrane, plasma membrane, endoplasmic reticulum, or Golgi, while a matrix or tegument layer links the inner capsid to the outer membrane. Non-enveloped viruses often assemble in the nucleus or cytoplasm and accumulate until cell lysis. Maturation steps such as protease-mediated cleavages in retroviruses convert non-infectious precursors into fully infectious particles by rearranging internal architecture. Release closes the factory doors. Enveloped viruses bud, acquiring their envelope as they exit. Some bud into internal compartments and depart by exocytosis, others bud directly at the plasma membrane. Budding can spare the cell initially, allowing persistent production. Non-enveloped viruses more often trigger lysis, releasing a burst of particles. Certain enveloped viruses also induce cell-cell fusion, forming multinucleated syncytia that permit spread without exposure to extracellular antibodies. Cell injury arises from several mechanisms: direct lysis, diversion of resources, membrane fusion, accumulation of toxic viral proteins, or immune-mediated destruction of infected cells. Microscopy may reveal inclusion bodies, viral factories, or aggregates whose location, nuclear versus cytoplasmic, can hint at the virus group. Interferon responses, induced by sensing of foreign RNA or DNA, establish an antiviral state in neighboring cells. In turn, many viruses encode antagonists that block sensing, degrade signaling intermediates, or shut off host translation to tilt the contest. Now separate two tempos of infection. A lytic infection is productive and brief, rapid replication, assembly, and release, culminating in cell death or shedding. A latent infection is quiet. The viral genome persists with limited expression, often as an episome in the nucleus or integrated into host DNA, while production is minimal or absent. Herpes viruses are archetypal. After a primary episode, genome persistence in specific neurons or lymphoid cells can last years, with episodic reactivation triggered by stressors such as ultraviolet light or local injury. Reactivation produces new rounds of productive replication along the original pathways of tropism. Persistence differs subtly. Some viruses maintain ongoing, low-level production for long periods without full quieting. Hepatropic viruses, for example, may persist with stable reservoirs of nuclear DNA templates. A brief clinical cameo brings the tempos into focus. An adult notes grouped, painful vesicles on the lip margin after days of fatigue and sun exposure, then spontaneous resolution as weeks pass. That pattern reflects neuronal latency with periodic reactivation and local productive replication. New neurologic symptoms, eye pain with visual changes, or severe headache and confusion in such a setting would be reasons to seek urgent professional care to evaluate for deeper involvement. The value for you is the mechanistic forecast. Latency shapes timing and triggers, while lytic bursts shape abrupt onset and shedding. A quiet reflection: when you imagine a factory keyed to a single script, where the script itself decides which room is used and which tools are needed, replication stops feeling mysterious. You already know the next move once you read the label. You leave the print room with one more question at the threshold. If cellular parcels borrow your machinery to copy themselves, what of organisms that bring their own nuclei, their own ribosomes, and build walls of chitin? Yeasts bud, molds branch, and some switch forms with temperature like a dimmer. What does that architecture change about disease and detection? You walk through a glass house at dusk, where threads climb trellises and round buds pinch off into new growth. Mycology is this greenhouse of forms: yeasts that bud, molds that branch, and dimorphic species that switch shape with temperature. Each architecture predicting habitat, laboratory clues, and clinical patterns you can read calmly. Begin with the common blueprint. Fungi are eukaryotes, cells with nuclei, and they build cell walls from chitin and glucans rather than bacterial peptidoglycan. Their membranes use ergosterol instead of cholesterol. Those two facts, glucan-rich walls and ergosterol membranes, explain why laboratory stains and future therapeutics target different places than in bacteria. Yeasts grow as single cells that reproduce by budding or fission. When buds elongate without fully separating, they form pseudohyphae, chains with constrictions at the junctions. Molds grow as multicellular filaments called hyphae. When hyphae mesh into a mat, the structure is a mycelium. Septate hyphae contain cross walls with pores. Non-septate coenocided hyphae appear broad and ribbon-like. Many pathogenic fungi are dimorphic. At environmental temperatures they grow as molds. In mammalian tissues they appear as yeasts or specialized spherules. Mold in the cool, yeast in the warm captures the shift. Keep the greenhouse image as you examine reproductive structures. Asexual spores dominate clinical labs. Colonidia are asexual spores that form on specialized hyphae. In Aspergillus, chains of colonidia arise from a vesicle atop a colonidiophore, a tidy column you can sketch from memory. Sporangiospores, in contrast, sit enclosed in a sac, sporangium, at the tip of a sporangiophore, as seen in Mucorales, the group including Rhizopus and Mucoro. These distinct silhouettes, beaded chains versus a sac, become quick visual cues under the microscope. Translate architecture into clinical habits. Yeasts behave like moisture-loving shrubs. They colonize mucosa and invade bloodstream when barriers fail. Candida species colonize the oropharynx, gut, and vagina. In tissue they form budding yeasts and pseudohyphae that penetrate superficially. In the bloodstream, Candida can seed organs and indwelling devices, favoring surfaces where biofilms anchor. Cryptococcus neoformans and its relatives carry thick polysaccharide capsules. They prefer lungs and central nervous system in susceptible hosts, with a predilection for meningeal spaces. Molds act like climbing vines, thriving along air-exposed surfaces and avascular spaces. Aspergillus species produce septate hyphae that branch at acute angles and can colonize pre-existing lung cavities, forming fungus balls that move with position. In hosts with profound neutropenia, markedly low neutrophil counts, Aspergillus can invade blood vessels, causing infarction and nodular lesions. Mucorales grow as broad, non-septate hyphae with right-angle branching, invading nasal passages and sinuses. In susceptible hosts with high available iron, uncontrolled hyperglycemia, or neutropenia, they can penetrate tissue planes and vascular channels at alarming speed. Dimorphs link geography to form. Histoplasma capsulatum lives in soil enriched by bird or bat droppings. Inhaled microconidia convert to small oval yeasts and macrophages. Blastomyces dermatitis produces thick-walled yeasts with broad-based budding in tissue. Coccidioides grows as a mold in arid soils. In tissue it forms spherules packed with endospores. Paracoccidioides in Latin America yields multipolar budding yeasts, often described as a ship's wheel. Sporothrix schenckiae, associated with plant thorns and sphagnum, produces cigar-shaped yeasts in tissue and tracks along lymphatics after inoculation. Now anchor your greenhouse tour in the laboratory. Direct microscopy is the fastest first look. A potassium hydroxide KOH preparation dissolves keratin and clears skin or nail samples so you can see hyphae. Chalcofluor white binds chitin and cellulose, fluorescing fungal walls brightly. In tissue sections, silver stains such as gomorimethionamine, silver outline walls sharply. Periodic acid Schiff highlights polysaccharides. For encapsulated yeasts, negative contrast preparations, historically India ink, can show a clear halo around the cell. In practice, antigen detection for Cryptococcus in cerebrospinal fluid or serum is far more sensitive. Culture on Saborodextrose agar favors fungal growth by low pH and simple nutrients, but growth can be slow. Temperature phase testing 25 telopters versus 37 telopters versus 37 telopters versus 37 telopters. Beyond morphology, targeted antigen assays add specificity. Detection of galactomannan, a cell wall component of Aspergillus, aids early inference in high-risk hosts. 120-beta-D-glucan, a pan-fungal wall polymer, signals many invasive infections but spares organisms with minimal glucan exposure. Molecular tools, polymerase chain reaction (PCR), and mass spectrometry profiles such as matrix-assisted laser desorption, ionization time of flight (MLI-TOF) accelerate identification from colonies or sometimes directly from specimens. Clinical patterns follow immune ecology. Neutrophils, white blood cells that engulf and kill, are central to mold defense. Profound neutropenia or impaired oxidative burst increases risk for invasive aspergillosis or mucormycosis. T lymphocytes orchestrate control of dimorphic fungi and Cryptococcus. Deficits in cell-mediated immunity predispose to disseminated disease and meningoencephalitis. Breaches in cutaneous barriers invite Sporothrix and Candida locally. Breaches in mucosal barriers or intravascular devices invite candidemia. Pre-existing lung cavities beget aspergillomas. Asthma with sensitization can evolve into allergic bronchopulmonary aspergillosis, where immune responses to airway colonizing molds drive exacerbations. Dermatophytes, Trichophyton, Microsporum, Epidermophyton, prefer keratin and remain superficial, producing annular scaly plaques, tinea on skin, hair, or nails. Wood's lamp ultraviolet light can highlight fluorescence in some Microsporum species on hair. KOH scrapings show septate hyphae within stratum corneum. Onychomycosis, fungal nail infection, thickens and distorts nails. Diagnosis still rests on microscopy and culture because nail dystrophies have many causes. Bring the patterns together with silhouettes you can recall instantly. Candida albicans, budding yeasts with pseudohyphae, a warm serum germ tube test. Short hyphal outgrowths from yeasts has traditional use in the lab. Aspergillus, acute angle branching, septate hyphae. Radiology may show nodules with a halo in neutropenic hosts. Mucorales, broad posiseptate hyphae with right angle branching. Black necrotic eschars in the nasal cavity or palate can appear as tissue ischemia advances. Cryptococcus, encapsulated yeasts with a mucopolysaccharide halo. Central nervous system involvement presents with subacute headache and elevated opening pressure. Coccozetioids, spherules with endospores. Desert exposure and arthralgias align with primary disease. Blastomyces, broad-based budding yeasts. Cutaneous verrucous lesions can accompany pulmonary disease. Histoplasma, tiny intracellular yeasts within macrophages. Exposure to caves or roosts fits. Paracoccozetioids, multiple buds radiating from a thick mother cell. Sporothrix, nodular lymphangitic spread from inoculation sites. A measured clinical vignette studies the map. A gardener pricks a finger on a rose thorn and over days notes a painless papula that ulcerates. Weeks later, a trail of nodules develops ascending the forearm along lymphatic channels. The exposure to plant material, the chronic course, and the beaded line suggest lymphocutaneous sporotrichosis. New fever, rapidly spreading redness with severe pain, or signs of systemic illness would be cues to seek professional care to evaluate depth and spread. The lesson is pattern recognition, route plus tempo plus silhouette, narrow possibilities before specialized tests confirm. A short, quiet thought. When you treat each fungal form as a plant with a favorite bed, yeast shrubs, hyphal vines, shapeshifters, you replace memorization with a garden plan. The paths become familiar, surprises become fewer. Before you leave the greenhouse, glance toward a neighboring conservatory where life cycles lengthen and vectors land at dusk. Some eukaryotes do not simply bud or branch, they divide in hosts and develop in insects, crossing borders with complex timing. What changes when the organism carries its own itinerary through protozoan stages and helminth forms, and a bite or a cyst begins the story? You imagine a map stretched across continents, marked with rivers, forests, and insects that flit from host to host. Parasitology is this cartography. Protozoa and helminths, whose life cycles cross human, animal, and vector domains. Unlike bacteria and fungi, parasites are eukaryotic organisms with complex developmental stages, often involving multiple hosts or environmental reservoirs. Their ability to shift forms and habitats explains persistence, transmission, and clinical patterns that can otherwise seem bewildering. Begin with protozoa, the single-celled travelers. They reproduce by binary fission and often cycle through trophozoite, active feeding, and cyst, dormant, resistant forms. Giardia lamblia is an example. Trophozoites attach to intestinal villi with a ventral disc, disrupting absorption, while hardy cysts survive in water to transmit infection. Entamoeba histolytica forms invasive trophozoites that penetrate colonic mucosa, causing flask-shaped ulcers, while cysts spread via contaminated hands or food. Trichomonas vaginalis lacks a cyst form. It transmits directly by intimate contact as motile trophozoites. Apicomplexans, protozoa with specialized apical complexes for cell invasion, illustrate the elegance and difficulty of parasitology. Plasmodium species transmitted by Anopheles mosquitoes cause malaria through cycles of hepatic schizogony, asexual multiplication in liver cells, and erythrocytic schizogony in red blood cells. Periodic rupture of infected erythrocytes releases merozoites, explaining rhythmic fevers. Toxoplasma gondii alternates between sexual reproduction in cats and asexual stages in intermediate hosts, including humans, where tissue cysts persist silently in muscle and brain. Cryptosporidium species produce acid-fast oocysts that resist chlorine, allowing waterborne outbreaks. Their trophozoites remain in epithelial brush borders, provoking watery diarrhea. Flagellated protozoa illustrate vector-borne spread. Trypanosoma brucei, transmitted by tsetse flies in sub-Saharan Africa, shifts surface glycoproteins to evade immunity, producing waves of parasitemia and, eventually, central nervous system invasion, African sleeping sickness. Trypanosoma cruzi, transmitted by triatomine kissing bugs in the Americas, enters cells, transforms into a mastogotosin, damages myocardium and gut neural plexuses, leading to chronic cardiomyopathy and megasyndromes. Leishmania species, spread by sandflies, cycle between extracellular promastogotes in the vector and intracellular amastogotes in human macrophages, producing cutaneous ulcers, mucocutaneous lesions, or visceral disease depending on species and host response. Helminths are multicellular, elongate organisms, worms with specialized organ systems. Nematodes, roundworms, are cylindrical, non-segmented, and separate sexes. Ascaris lumbricoides matures in the intestine but migrates through lungs during development, explaining cough and eosinophilia before intestinal obstruction. Hookworms penetrate skin as larvae, travel via blood to lungs, ascend airways, are swallowed, and mature in the intestine, where they attach and feed, causing iron deficiency anemia. Enterobius formicularis, pinworm, lays eggs around the anus at night, causing perianal itching and autoinfection cycles. Strongyloides, Starcoralis, can complete both free-living and parasitic life cycles in immunosuppressed hosts. Autoinfection may accelerate into hyperinfection. Cestodes, tapeworms, are flat, segmented helminths. The head scolex bears suckers or hooks for attachment. Segments proglottids contain reproductive structures. Taenia saginata and Taenia solium mature in human intestines after ingestion of larval cysts from undercooked beef or pork. T. solium eggs ingested directly can develop into larval cysticerci in tissues, including brain, causing neurocysticercosis with seizures. Diphyllobothrium latum, the fish tapeworm, can cause vitamin B12 deficiency by absorbing the nutrient in the small intestine. Trematitodes, flukes, are flat, leaf-shaped helminths with suckers. They require snails as intermediate hosts. Schistosoma species penetrate skin directly from freshwater. Adults reside in venous plexuses, producing eggs that elicit granulomatous inflammation in intestines, liver, or bladder. Chronic schistosomiasis can lead to portal hypertension or bladder carcinoma. Other trematitodes, such as Clonorchis sinensis, liver fluke, acquired from raw freshwater fish, reside in bile ducts and can predispose to cholangiocarcinoma. Vectors are the couriers on this map. Mosquitoes transmit Plasmodium, Wuchereria, lymphatic filariasis, and arboviruses. Sandflies transmit Leishmania. Tsetse flies carry Trypanosoma brucei. Triatomine bugs spread Trypanosoma cruzi. Black flies transmit Onchocerca volvulus, the agent of river blindness. The geography of vectors dictates endemic zones, and vector control measures reshape epidemiology as much as therapeutics. Immune response shapes outcome. Helminths often provoke eosinophilia, elevated eosinophil counts through release of cytokines that recruit these cells to fight multicellular invaders. Protozoa that invade cells demand T-cell responses. Impaired cellular immunity permits severe dissemination. Chronic infections often represent a balance. Parasites suppress or modulate host responses enough to persist without killing the host outright, ensuring their own transmission. Anchor the map with a clinical vignette. A traveler returns from sub-Saharan Africa with cyclical fevers every 48 hours, chills and sweats. The pattern fits red blood cell cycles of Plasmodium vivax or P's ovale, where synchronous schizogony explains regular fever spikes. If altered mental status, severe anemia, respiratory distress, or jaundice were to appear, these would be cues for immediate professional evaluation to assess for severe malaria. For you, the clinical anchor is life cycle logic. Stage-specific replication translates directly into rhythmic symptoms. Pause with a short reflection. When you frame parasites as travelers with itineraries, cystotrophozoite, larva to adult, vector to host, you replace confusion with a timetable. Symptoms become markers of where the parasite is on its journey. You close the map by noticing that complexity in life cycles complicates laboratory detection. Some stages shed in stool, others in blood, others in tissue. What if you could instead interrogate the host directly, asking what antigens are present or what antibodies have arisen? The next step opens the diagnostic wing, beginning with how specimens are collected and cultured to reveal the invisible. You picture a quiet logistics hub at night. Sealed parcels arrive from distant neighborhoods, barcodes scanned, contents routed to the right benches, then into warm rooms where signals appear on schedule. Diagnostic microbiology begins long before plates and machines. It begins with the parcel, the specimen, and every decision you make upstream determines whether the lab receives a clear message or only noise. Define the message precisely. A specimen is material collected from a body site to answer a focused question about infection at that site. Pre-analytical variables, what you collect, how you collect it, the container, the temperature, and the time to the lab, govern the value of every downstream test. Contamination means adventitious organisms introduced during collection or from adjacent flora. Colonization means microbes inhabit a site without causing tissue damage. Infection means microbes replicate and provoke injury. The lab reads these distinctions indirectly by quantity, organism identity, and context. Your job is to send a message that lets them tell which is which. Think of site-specific messaging. Blood is a sterile compartment. Organisms detected there matter. Multiple sets increase the chance of detection and help separate true bloodstream infection from skin contaminants because the same organism in more than one set and with similar time to positivity patterns strengthens the signal. Cerebrospinal fluid, CSF, is also sterile. Prompt delivery at body temperature preserves fragile pathogens. Urine from the bladder can be sterile, but lower tract flora sit at the outlet. Clean catch techniques and rapid transport reduce noise. Sputum aims to sample lower airways, yet saliva from the oropharynx carries abundant commensals. Microscopic screening for epithelial cells versus neutrophils helps the lab judge whether the sample represents lung secretions or mouthwash. For wounds, deep aspirates from the advancing edge convey the true actors. Superficial swabs often capture bystanders. Stool contains a busy community. Targeted requests for enteric pathogens guide the lab to use selective media and appropriate incubation, rather than treating the sample as a general culture everything task. Now route the parcels onto media, the growth environments that translate metabolism into visible colonies. Non-selective plates such as tryptic soy agar with 5% sheep blood support a broad array of bacteria and reveal hemolysis patterns that carry clues. Enriched plates like chocolate agar, heated to release red cells and release factors, nurture fastidious organisms that require extra growth factors. Selective and differential plates add 2 layers of logic. Inhibitors suppress certain groups while color reactions report on specific pathways. A classic example is a bile salts-containing medium that inhibits gram-positives and differentiates lactose fermenters by acid-driven indicator shifts. Pale, oxidase-positive colonies from water-exposed devices may hint at non-fermenters, whereas pink colonies from stool suggest enteric fermenters. For obligate anaerobes, reducing media and oxygen-free chambers create the low redox environment necessary for growth. For microaerophiles, controlled oxygen and carbon dioxide mixtures simulate their natural niches. Some organisms demand highly specialized recipes: charcoal yeast extract with iron and cysteine for aquatic intracellular pathogens, selective media containing antibiotics to suppress flora when you seek a delicate slow-growing respiratory coccus. Timing is the clock on the wall of the hub. A direct gram stain from many specimens, CSF, joint fluid, aspirates, gives a same-day silhouette, color, shape, and arrangement that narrow choices immediately. For many common bacteria, visible growth appears by the next day. Subculture and biochemical or mass spectrometric identification follow, often delivering a name within another day. Susceptibility testing, exposing standardized inocula to antimicrobial concentrations on plates or in broths, adds more time. Some agents require enriched atmospheres or extended incubation. A curved fastidious gut organism may reveal itself only after days in microaerophilic jars. Mycobacteria and certain dimorphic fungi challenge patience. Their slow replication stretches culture to weeks, and specialized containment safeguards staff during handling. Blood cultures deserve a closer, conceptual look because they convert invisible replication into a machine-detected signal. Bottles contain nutrient broth and resin systems to neutralize inhibitory substances. Instruments monitor carbon dioxide or pressure changes to flag growth. Time to positivity. The interval between incubation and the alarm correlates with starting bacterial load and sometimes with clinical significance. Early positivity in multiple bottles from separate draws argues for true bacteremia. Very late, single bottle growth of skin flora leans toward contamination, though exceptions exist. When a bottle flags, a technologist performs a gram stain and subcultures to plates, coupling speed with specificity. Quality control threads through every step. Labels must match patients. Requisitions must pose answerable questions, evaluate for anaerobes or suspected enteric pathogen-focused choices. Transport temperature matters. Cooling preserves some organisms and cripples others. Oxygen exposure ruins strict anaerobes. Swabs trade convenience for volume and survival. Fluids and tissues outperform swabs by delivering more organisms and protecting anaerobes within clots. Each specimen type carries an ideal container and timeline. The closer you come to that ideal, the clearer the downstream story becomes. Interpretation is not a single event, but a reading of patterns over time. The first day produces microscopy clues, the second, colony morphology and preliminary identifications, the third, conformations and early susceptibility, the fourth and beyond, late bloomers and expanded panels. Parallel results from multiple sites increase confidence. The same gram-negative rod from blood and urine, both with compatible counts and timing, frames a coherent source. The same diphtheroid from a deep wound and from the skin surface may reflect contamination in one and true infection in the other, depending on quantity and context. A moment in the hub's call room anchors the abstractions. Late evening, a call reports gram-negative diplocracy in CSF from a patient with fever and neck stiffness. That silhouette in a sterile space has high predictive value for an encapsulated fastidious pathogen. Clock speed matters because delays risk outcomes. New confusion, a purpuric rash, or declining level of consciousness in such a scenario would be reasons for immediate professional care escalation. Your takeaway is not to memorize names now, but to see how specimen choice, sterility of site, and gram morphology combine to inform urgency while definitive culture proceeds. Broaden the lens to mixed infections and devices. In deep abscesses, mixed anaerobic flora are common. Drainage reduces pressure and restores oxygen gradients unfriendly to strict anaerobes, while cultures document participants. On indwelling catheters, biofilm communities shed planktonic cells intermittently. The same species recurring from line-drawn specimens points toward device involvement. When you see no growth but still suspect infection, timing and prior exposures matter. If antimicrobials were started before collection, viable counts may fall below detection. If the pathogen is intracellular or extremely fastidious, culture-independent methods may be needed. Finally, accept that culture is a conversation, not a verdict. Growth tells you who can thrive under the conditions you provided, not necessarily who initiated disease. Normal flora can overgrow on plates. Pathogens can hide behind slow tempo. This is why you correlate with site, symptoms, imaging, and inflammatory markers, building a convergent case rather than relying on any single line. A brief reflection. When you treat the lab like a well-run postal hub, right parcel, right address, right truck, the messages arrive legible. Noise reduces, confidence rises, decisions slow down in your mind, even as the clock moves. You step out of the hub to a parallel corridor where growth is not required at all. What if the message could be read from signatures, unique proteins on a microbe's surface, nucleic acid sequences, or your own antibodies marking an encounter? The next hall opens to antigen detection, nucleic acid amplification, and serology, where timing and targets reshape what positive means. You walk into a reading room where messages arrive without culture, barcodes of protein, strings of nucleic acid, and echoes of your own antibodies. These are rapid signatures that complement growth-based methods: antigen detection, nucleic acid amplification, and serology. Each answers a different question. Is a piece of the microbe present now? Is the microbe's genome here now? Or has your immune system met this agent recently or in the past? And each depends on timing, site, and context. Start with antigen detection, the direct barcode. An antigen is a molecule, often a protein or polysaccharide, recognized by antibodies. Tests use antibodies anchored on a surface to capture the target. A second labeled antibody creates a visible or measurable signal. Formats vary. Lateral flow immunoassays are disposable strips that give qualitative results within minutes at the point of care. Enzyme immunoassays or chemiluminescent immunoassays run in analyzers and yield quantitative or semi-quantitative readouts. Direct fluorescent antibody tests label targets with fluorescent tags and require microscopy. Strengths are speed, simplicity, and the ability to detect components that may persist when organisms are difficult to culture. Weaknesses are variable sensitivity, chance of missing true positives, and cross-reactivity, chance of false positives. Examples clarify use. In bacterial disease, urinary antigen tests detect polysaccharide fragments from encapsulated organisms. A classic pair targets a common respiratory coccus and a water-associated intracellular pathogen, providing rapid clues in severe community-acquired pneumonia. In invasive mycoses, soluble fungal wall components can be measured in serum or other fluids. A mannose-rich capsular antigen signals an encapsulated yeast, a galactofuranose polymer suggests a septate mold, and a 3-butyglucan indicates many but not all fungi. Each has caveats. Some antibiotics or blood products can yield false positives. Some fungi produce low amounts of detectable polymers in blood. In parasitology, rapid malaria tests detect histidine-rich protein 2 or parasite lactate dehydrogenase (PLDH) in blood. Deletions in the HRP2 gene in some regions reduce HRP2 test performance, so pairing with PLDH-based assays can help. Move to nucleic acid amplification tests. NAT-Seq, the string reader. A NAT-Seq is any method that amplifies a target nucleic acid sequence to detectable levels. The most common is polymerase chain reaction (PCR), which cycles through heating and cooling to denature DNA, anneal primers, and extend new strands, doubling target copies each cycle. Reverse transcription PCR (RT-PCR) first converts RNA to complementary DNA before amplification. This is used for RNA viruses and some transcripts. Isothermal methods, such as loop-mediated amplification, run at a constant temperature and can be engineered into rapid, portable formats. Multiplex platforms amplify many targets simultaneously, useful for panels that survey dozens of respiratory or gastrointestinal pathogens in one run. NAT strengths are sensitivity, speed after extraction, and specificity when primers and probes are well chosen, but interpretation requires discipline. Detection of a genome does not equal disease. It confirms presence above a threshold. For organisms that colonize without causing injury, a positive NAT from a non-sterile site may reflect carriage rather than causation. Cycle threshold CT values in real-time PCR represent the cycle at which fluorescence crosses a set level. Lower CT suggests more starting material, but CT is not interchangeable across platforms or specimens and should not be overinterpreted as a direct measure of severity. In Clostridioids difficile testing, for example, NAT detects toxigenic strains by their toxin genes, yet clinical disease hinges on active toxin production. Algorithms often pair glutamate dehydrogenase antigen screening with toxin immunoassays and reserve NAT as an adjudicator to avoid overcalling colonization. Dense can also detect resistance-associated genes. A methicillin resistance determinant, a penicillin-binding protein variant, marks a staphylococcal lineage as resistant to beta-lactamase. Stable penicillins, vancomycin resistance elements in enterococci predict glycopeptide resistance. Carbapenemase genes in gram-negative rods forecast broad beta-lactam resistance. Rapid detection from positive blood culture bottles accelerates infection control decisions and helps prioritize conceptual options while awaiting full phenotypic susceptibility data. For mycobacteria, cartridge-based NATs amplify specific sequences and probe common resistance mutations, compressing weeks of culture into hours for initial guidance, while culture still proceeds for comprehensive susceptibility and epidemiology. Now read serology. the echo in your immune archive. Serology detects antibodies your immune system generates after exposure. Immunoglobulin M, IgM, usually appears first and wanes. Immunoglobulin G, IgG, appears later and persists, sometimes for years. Immunoglobulin A, IgA, can mark mucosal responses. The timing of sampling relative to symptom onset determined ability. In early illness, antibodies may be absent. The window period. Paired serology, comparing acute and convalescent samples weeks apart, looks for a significant rise in antibody levels, supporting recent infection. Avidity testing measures the strength with which antibodies bind. Low avidity IgG suggests recent primary response, while high avidity IgG suggests a more remote infection. Neutralization assays, which assess an antibody's ability to block infection of cells in culture, can add functional confirmation in specialized settings. Serology shines for agents that are difficult to culture or detect directly after the acute phase. In some spirochetal infections, two-stage algorithms use a specific assay to detect pathogen-directed antibodies and a non-specific assay to track activity via titers. In hepatropic viruses, antigen-antibody patterns map phases. A surface antigen indicates current presence, core antibodies track exposure, and surface antibodies mark immunity. Combinations indicate acute infection, past infection, or vaccination. Fourth-generation combination assays that detect both antigen and antibody reduce the diagnostic window in certain retroviral infections. Yet serology falters in immunocompromised hosts with blunted antibody responses and can be confounded by cross-reactive antibodies in related organisms. Interpret results through the lens of pretest probability. Sensitivity and specificity are test properties. Positive and negative predictive values are context properties that shift with disease prevalence and clinical suspicion. In low prevalence settings, false positives loom large. In high prevalence settings, false negatives matter more. A result does not stand alone. It joins symptoms, exposure history, imaging, and other labs in a Bayesian update. Reproducibility across specimen types strengthens confidence. A respiratory panel NAIT identifying a pathogen in a nasopharyngeal swab and a compatible antigen in lower airway fluid tells a coherent story. Specimen quality still rules. For antigen detection and NAT, inhibitors in specimens can block reactions and poor sampling misses targets. Nasopharyngeal swabs placed too shallowly reduce yield. Sputum contaminated with saliva dilutes signal. Blood collected in inhibitory tubes can impede amplification. Internal controls embedded in assays help flag inhibition. Storage and transport matter. Nucleic acids degrade with time and temperature. Antigens denature or disperse. Antibodies remain stable but require the right tube and separation steps. A composed clinical cameo locks the principles. An older adult with fever, cough, and hypoxemia is admitted in winter. Within hours, a multiplex respiratory NAT detects a single enveloped RNA virus in a nasopharyngeal swab. An antigen test on lower respiratory fluid aligns. Bacterial cultures remain negative. The concordant direct detections and the sterile blood cultures focus attention on supportive care and secondary complications. If instead the NAT were positive for a colonizing bacterium from a superficial swab, while blood cultures flagged a different gram-negative rod from two sets, the weight would shift toward the bloodstream isolate as the prime actor. New confusion, rapidly progressive breathing difficulty, or chest pain would be reasons to seek urgent professional evaluation. The lesson is triangulation, site timing, and concordance dictate confidence. A brief reflection: when you treat tests as structured questions, is it here now versus was it here recently, the noise subsides. Answers become coordinates, not verdicts, and you navigate with steadier hands. You step out of the reading room into a corridor where knowledge about walls and ribosomes will soon matter. Now that you can name the microbe and read its signatures, how do you conceptually target peptidoglycan without harming yourself, or quiet a 50S or 30S machine without silencing your own? The next section opens the pharmacology workshop, where structure predicts selectivity. You step into a quiet workshop where locks and gears lie open on the bench, each inviting a precise tool. Antibacterial pharmacology is this bench. Choose the tool that fits a bacterial part you do not possess. Apply it with the right rhythm, and the machine stops while yours keeps turning. Selectivity rests on two separations you already know. Bacteria build peptidoglycan cell walls that you lack, and they translate proteins on 70 ribosomes unlike your 80 ribosomes. Tonight you handle both realms, walls and ribosomes, and watch how that choice shapes speed, spectrum, and resistance. Begin at the wall with the beta-lactams, the socket set most often reached. A beta-lactam ring mimics the terminal dipeptide at peptidoglycan crosslinks. Penicillin-binding proteins, PBPs, the enzymes that knit peptide bridges, mistake the drug for their substrate, form a covalent adduct, and stall transpeptidation. The lattice fails to mature, autolytic enzymes tip the balance toward lysis. Killing is classically bactericidal and time-dependent. Efficacy rises as concentrations stay above a minimal inhibitory threshold over the dosing interval rather than by high peaks. Access matters. In gram-positives, PBPs reside just beneath a thick but accessible wall. In gram-negatives, an outer membrane and porin's gate entry, so porin repertoire and periplasmic beta-lactamases strongly influence activity. Subclasses tune fit and stability. Some handle common beta-lactamases better, some traverse particular porins more efficiently, some bind altered PBPs with higher affinity. Resistance mechanisms align with the blueprint. Enzymes that hydrolyze the ring beta-lactamases altered targets that reduce binding, e.g. a PBP variant, decreased entry via porin loss, and accelerated efflux. The logic is mechanical. If the wrench cannot reach or the bolt head has changed shape, torque never transfers. Move one shelf to glycopeptides, the large clamps. These agents bind the D-ala-de-ala termini of peptidoglycan precursors, sterically blocking both transglycosylation, polymerization of glycan strands, and transpeptidation cross-linking. Because the target lies outside the membrane, access to gram-positive walls is straightforward. The outer membrane of gram-negatives, however, excludes these bulky molecules, explaining their limited range. A key adaptive escape replaces the terminal D-ala with D-lac or D-ser, reducing binding affinity by altering a single hydrogen bond pattern. Small chemical edits at the wall's tip defeat a large clamp. Conceptually, killing is concentration exposure dependent, and distribution reflects size. Deep tissues are reachable, but penetration into certain privileged spaces improves when inflammation opens barriers. Infusion-related reactions and kidney handling are part of safety considerations, yet the educational point remains mechanism, a direct pinch at the wall's handle. Shift now from masonry to the factory floor. Translation on 70S ribosomes. Bacterial ribosomes comprise a 30S small subunit and a 50S large subunit. Their rRNA and proteins differ enough from yours to allow selective binding. In general, wall agents are bactericidal by structural catastrophe, while many ribosome agents are bacteriostatic by slowing protein synthesis. There are notable exceptions you will see. Macrolides are stoppers at the 50 exit tunnel. They bind 23S rRNA in the large subunit near the peptidyl transferase center and block translocation, the step in which the ribosome advances the mRNA-tRNA complex. The nascent peptide cannot exit, elongation stalls. Because transport into host cells is favored and intracellular concentrations become high, macrolides reach pathogens that reside within phagocytes and epithelial cells, aligning with respiratory and atypical niches. Resistance follows three mechanical routes: methylation of the 23S binding site, which reduces drug affinity, active efflux pumps that lower intracellular levels, and point mutations that distort the pocket. The class effect is usually bacteriostatic, and pharmacodynamics favor maintaining exposure over time rather than chasing high peaks. If you picture the tunnel as a corridor, macrolides place a firm barrier a few steps in. Tetracyclines sit earlier in the 30th choreography. They reversibly bind the small subunit at the A site, preventing aminoacyl tRNA from docking with the codon. Without an incoming charged tRNA, elongation cannot proceed. Their chelating structure gives them broad entry into cells and unusual tissue distribution, reaching intracellular bacteria and diverse compartments. Resistance often arises through efflux pumps that export the drug or ribosomal protection proteins that dislodge it, restoring A-site access despite drug presence. Classically bacteriostatic, they temper growth across a wide range of organisms. Their interactions with divalent cations, deposition and developing calcified tissues, and photosensitivity are well known in safety discussions, but the mechanistic memory hook is enough. They keep the A site empty. Aminoglycosides, though also 30S binders, change the tempo and the outcome. They interact at the decoding site, inducing conformational changes that cause misreading of codons and incorporation of incorrect amino acids. Defective proteins accumulate, some embed in the membrane and increase permeability, and a vicious cycle of uptake and damage ensues. The result is rapid, concentration-dependent bactericidal activity with a post-antibiotic effect, continued suppression after levels fall below simple inhibitory thresholds. Uptake across the inner membrane requires an oxygen-dependent transport step and a favorable membrane potential, which is why activity is poor in strict anaerobic conditions and can be blunted in acidic hypoxic foci. Synergy with wall-active agents emerges directly from mechanics. If the wall is nicked open, entry improves and ribosomal targeting accelerates. Enzymatic modification, acetylation, phosphorylation, adenylation of the drug, target site mutations, and reduced transport are common resistance routes. Safety concerns concentrate in kidneys and inner ear because their transporters and mitochondrial ribosomes present vulnerabilities. Again, a reminder that selectivity is relative, not absolute. A few cross-cutting principles steady your hand at the bench. First, time versus concentration matters. Beta-lactams and macrolides usually reward sustained exposure above a threshold. Aminoglycosides reward high peaks with rest periods, reflecting different kill kinetics and adaptive responses. Second, access governs apparent spectrum as much as intrinsic binding. Outer membranes, porins, efflux pumps, and intracellular residency all shape who is reachable. Third, target edits rarely cost the bacterium nothing. Altered PBPs can slow wall assembly. Methylated ribosomal pockets can reshape fitness under some conditions, trade-offs that influence which variants persist when pressure eases. Now translate tools back to scenes you recognize. In a prosthetic joint infection with biofilm architecture, slow-growing cells in a matrix tolerate exposures differently from actively dividing planktonic counterparts. Wall agents still matter, but ribosome agents may penetrate cells better, and combinations can exploit complementary access. In a deep abscess with low pH and low oxygen, aminoglycoside entry falters mechanistically. Draining the space alters chemistry as much as any drug choice could. In a persistent intracellular respiratory infection, agents that concentrate within macrophages align with the niche. In a rapidly progressive bloodstream infection with high bacterial burden, fast bactericidal wall disruption carries conceptual appeal. These are not prescriptions, they are maps from mechanism to context. A brief clinical cameo keeps caution close. A patient with fever, flank pain, and chills becomes lightheaded. Blood cultures later reveal a gram-negative rod from more than one set. Hypotension, confusion, or decreased urine output in such a presentation would be reasons to seek immediate professional care to evaluate for sepsis and organ perfusion. Your lesson here is not which specific agent to choose, but why time-dependent wall blockers, outer membrane access, and periplasmic enzymes loom large in early thinking while definitive testing proceeds. A quiet reflection: when you picture a wrench on a bolt or a stopper in a tunnel, the fog lifts. You do not memorize lists. You match tools to parts and tempo, and the machine tells you what will and will not budge. You set the wall and ribosome tools back on the bench and glance at a different drawer where the gears are genomic. What if you halt DNA supercoiling, jam an RNA polymerase mid-track, or starve the folate pathway that supplies bases altogether? And how do bacteria reroute around those blocks overnight? You open the next drawer in the workshop and find finer instruments, tools that slip into coils of DNA, halt transcription, or block the assembly line of nucleotide synthesis. These agents target processes downstream of walls and ribosomes: replication, transcription, and metabolism. Their selectivity arises because bacterial enzymes for these steps differ subtly but meaningfully from yours, and those small distinctions allow intervention without shutting down your own genome factories. Begin with DNA gyrase and topoisomerase IV, enzymes that manage DNA supercoiling. Bacterial chromosomes are circular and must be compacted into the nucleoid. During replication, unwinding generates torsional stress. DNA gyrase, a type II topoisomerase, introduces negative supercoils to relieve that stress, while topoisomerase IV separates daughter chromosomes after replication. Quinolones, such as ciprofloxacin and its relatives, bind the DNA enzyme complex and freeze it in place, creating double-strand breaks the bacterium cannot repair. The result is bactericidal action. Gram-negative organisms rely more heavily on gyrase, gram-positives on topoisomerase IV, but in practice both enzymes can be targets. Resistance emerges through stepwise mutations in the quinolone resistance-determining regions of gyrase and PARC genes, as well as plasmid-mediated proteins that protect the enzymes or efflux pumps that lower intracellular drug concentrations. Mechanistically, killing is concentration-dependent, with efficacy tied to the ratio of drug peak to minimal inhibitory concentration. Conceptually, the drug wedges into the gears of DNA topology, and the machine locks. Move to RNA polymerase, the enzyme that transcribes DNA into RNA. Rafamicins, rifampin foremost, bind the beta subunit of bacterial RNA polymerase near the active center and prevent elongation after initiation. By blocking the channel for nascent RNA, rifampin halts transcription. The result is bactericidal against many rapidly dividing bacteria, because it penetrates biofilms and intracellular space as well. Rifampin has special value when bacteria hide in niches or foreign materials. Resistance, however, can arise quickly from single-point mutations in the ARPOB gene encoding the binding site, which is why rifampin is rarely used alone. The conceptual image is a cork in the barrel. The polymerase starts but cannot extend and production stalls. Now step into metabolism, specifically folate synthesis. Folate is essential for building purines and thymidine, the bases of DNA. Humans obtain folate from diet, but bacteria must synthesize it from precursors. Sulfonamides are structural analogues of para-aminobenzoic acid, PABA, a precursor in the pathway. They compete with PABA for dihydropteroate synthase, preventing folate synthesis at the first committed step. Trimethoprim targets dihydrofolate reductase, the enzyme that reduces dihydrofolate to tetrahydrofolate, blocking the final conversion to the active cofactor. Alone, each step can be bypassed or compensated. Together, sulfonamide plus trimethoprim form a sequential blockade, producing synergy. The combined effect is bactericidal against many susceptible organisms, though individually they are bacteriostatic. Resistance emerges from increased production of PABA, altered enzymes with lower drug affinity, or plasmid-encoded alternative reductases. Picture the pathway as a canal with two locks. Closing both prevents passage entirely, while closing only one allows leaks around. Mechanisms of selectivity remind you why these targets work. Your own DNA gyrases differ from bacterial ones in structure and are not bound by quinolones at therapeutic levels. Your RNA polymerase has different binding surfaces from bacterial polymerase. Your folate needs are met by dietary uptake. You do not use dihydropteroate synthase at all, so sulfonamides have nothing to inhibit. The differences are subtle but decisive. Tie back to clinical situations where these mechanisms matter. In urinary tract infections caused by gram-negative rods, quinolones concentrate well in urine and tissues, aligning with the pathogen's reliance on DNA gyrase. In prosthetic device infections where bacteria form biofilms, rifampin's ability to penetrate matrices and reach intracellular reservoirs makes it conceptually valuable in combination regimens, though never alone due to resistance risk. In certain parasitic infections, the folate pathway similarity is exploited by the same sulfonamide trimethoprim logic, yet all these uses rely on the mechanistic insight. DNA supercoiling, RNA elongation, or folate metabolism is jammed. A vignette anchors the gears. A patient treated with broad-spectrum antimicrobials develops new fever and abdominal discomfort. Cultures yield a toxin-producing spore-forming anaerobe. In such scenarios, DNA targeting or folate targeting drugs may not be effective because the organism is resistant by structure or pathway. Here the takeaway is that knowing mechanism prevents misplaced trust. If the target enzyme is absent or shielded, the tool will fail. Severe diarrhea with dehydration, blood, or abdominal rigidity in this context would be reasons to seek immediate professional care. Reflection, brief and calm. When you picture locks on DNA, corks in polymerases, and double gates on folate canals, you understand why inhibition works and why it sometimes fails. Mechanisms are not trivia, they are maps to where the levers sit. You close the drawer and glance at the next one labeled Eukaryotic Invaders. The tools inside differ, for they must pierce ergosterile membranes, poke at beta-glucan walls, or disturb protozoan DNA without harming you. What changes when the target is a fungus or parasite with organelles more like your own? You lift the lid of the next drawer and find heavier tools, ones designed not for simple prokaryotic machinery, but for eukaryotic invaders whose architecture overlaps with your own. Antifungals and antiparasitics must be more selective, targeting fungal ergosterile membranes, glucan walls, or parasite-specific pathways without striking your cells. The challenge is sharper. Fungi and parasites are closer relatives to you than bacteria are, so the space for selective toxicity is narrow and precious. Begin with antifungals that strike the membrane. Polyenes such as amphotericin B bind ergosterol, the principal sterol in fungal membranes. By inserting into the lipid bilayer and forming pores, they cause leakage of ions and metabolites leading to cell death. Selectivity comes from amphotericin's higher affinity for ergosterol than for your cholesterol, though collateral interaction with host membranes explains toxicity. Polyenes are fungicidal, and their broad activity spans yeasts, molds, and dimorphs. The conceptual image is a crowbar prying open tiles in the fungal membrane, leaving holes through which the cell's contents spill. Azoles, imidazoles, and triazoles target the ergosterol synthesis pathway by inhibiting linosterol 14-alpha demethylase, a cytochrome P450 enzyme. This blockage prevents conversion of linosterol to ergosterol, resulting in depleted ergosterol and accumulation of toxic intermediates that disrupt membrane structure. Azoles are generally fungistatic, though against some fungi and in high concentrations they can be fungicidal. Because the target is a cytochrome P450, interactions with your own P450 system complicate therapy, but the selective difference in enzyme isoforms underlies their utility. Resistance mechanisms include upregulation of efflux pumps, mutations in the target enzyme, and pathway bypasses. Think of azoles as removing a key roof tile. Without it, the roof warps and leaks. Echinocandins strike the wall, not the roof. They inhibit BET123D-glucan synthase, an enzyme essential for synthesizing a major polysaccharide of fungal cell walls. Without glucan, the wall weakens, osmotic fragility increases, and the fungus lyses. These agents are fungicidal against many yeasts and fungistatic against most molds. Selectivity rests on your lack of a glucan wall. Your cells are untouched by this mechanism. Resistance can develop through mutations in the FKS genes encoding glucan synthase subunits. Picture the echinocandin as a saw cutting through a scaffold beam. The fungal wall collapses under pressure. Other antifungals target nucleic acid synthesis. Flucytosine, a fluorinated pyrimidine analog, enters fungal cells via a cytosine permease and is converted by fungal cytosine deaminase to 5-fluorouracil, which inhibits thymidylate synthase and disrupts DNA and RNA synthesis. Humans lack cytosine deaminase, so selectivity derives from fungal activation of the drug. Resistance emerges quickly if used alone, so flucytosine is typically combined with polyenes or azoles. Conceptually, flucytosine is a disguised letter slipped into a script, causing garbled instructions. Shift to antiparasitics. Their diversity reflects the diversity of parasites. Protozoa and helminths differ profoundly, so strategies vary. For protozoa that thrive in low-oxygen niches, metronidazole offers a focused mechanism. Inside anaerobic or microaerophilic organisms, nitroreductase enzymes reduce metronidazol's nitro group, generating reactive intermediates that damage DNA. Your aerobic cells lack these enzymes, so selectivity holds. Against protozoa such as Entamoeba histolytica, Giardia lamblia, and Trichomonas vaginalis, metronidazole is potent and directly linked to their unique metabolism. The metaphor is a candle lit only in oxygen-poor rooms, burning paper there but staying dark elsewhere. Helminth therapy targets unique structures. Benzimidazoles, such as albendazole, bind to parasite B-tubulin and inhibit microtubule polymerization, disrupting glucose uptake and cell division. Your tubulin differs sufficiently to allow selectivity, though overlap explains side effects at high exposure. Paralytic agents exploit worm neuromuscular physiology, some open chloride channels gated by glutamate, others activate or inhibit nicotinic acetylcholine receptors. The worms are immobilized and expelled by peristalsis. Praziquantel, used for trematodes and cestodes, increases calcium permeability of parasite membranes, causing tetanic contraction and dislodgement. Its exact selectivity relates to differential calcium handling in worms versus humans. These strategies illustrate how exploiting neuromuscular or structural peculiarities unique to worms frees them from your tissues without harming your own system substantially. Resistance arises in parallel ways. For fungi, target alteration, efflux, and metabolic rerouting limit antifungal effectiveness. For protozoa, reduced drug activation or enhanced efflux blunt metronidazol's DNA damage. For helminths, mutations in tubulin genes alter benzimidazole binding, and changes in ion channels reduce responsiveness to neuromuscular blockers. Resistance spreads more slowly than in bacteria because fungi and parasites reproduce less rapidly, yet it still shapes epidemiology and treatment landscapes. Now anchor the mechanisms in clinical frames. A patient with persistent fever, cough, and nodular infiltrates after prolonged neutropenia is suspected to have invasive aspergillosis, and echinocandin's wall-directed action may steady the course, but tissue invasion and angioinvasion highlight limits of fungostatic activity. A traveler with prolonged diarrhea after untreated water exposure has trophozoites in stool consistent with giardia. The low oxygen metabolism makes metronidazole effective conceptually. A child with an itchy perianal rash at night is diagnosed with pinworm. A benzimidazole's microtubule disruption aligns with the parasite's reliance on glucose uptake. In each case, mechanism directs tool choice. Severe persistent symptoms such as high fever, weight loss, blood in stool, or neurologic signs in these settings would be reasons to seek professional care promptly. A quiet reflection. When you see fungal walls as scaffolds of glucan, membranes as ergosterol roofs, and parasites as organisms with unique engines or nerves, the strangeness softens. Mechanisms become stories you can follow step by step, not opaque lists. You return the heavy tools to their drawer and notice a shield leaning against the wall marked resistance. Despite precise tools, microbes adapt. Enzymes destroy rings, pumps expel drugs, altered channels refuse binding. Next, you study how those shields rise and how misuse sharpens them further. You face the shield leaning in the corner. Resistance is that shield, bacteria altering locks, pouring solvents, or reshaping gates so your tools fail to bite. It is not one mechanism but many, each tied directly to the structures and processes you studied. The shield grows heavier when use is careless. It cracks more easily when you understand its seams. Start with enzymatic destruction. Beta-lactamases are the classic cutters. Enzymes secreted into the periplasm or beyond that hydrolyze the beta-lactam ring, neutralizing penicillins, cephalosporins, and related agents. Variants span from narrow-spectrum enzymes against early penicillins to extended-spectrum beta-lactamases (ESBLs) that cleave third-generation cephalosporins to carbapenemases that dismantle carbapenems, the most resilient beta-lactams. Some are plasmid-encoded and spread readily, others are chromosomal and inducible. Conceptually, they are scissors waiting at the gate, severing tools before they touch the wall. Next are altered targets. A single amino acid substitution in a penicillin-binding protein, PBP, can reduce beta-lactam affinity. A wholesale replacement, such as the acquisition of a variant PBP, confers broad resistance. Ribosomal binding sites can be methylated by enzymes, as with macrolides, or specific nucleotides can be mutated, as with aminoglycosides and tetracyclines, diminishing fit. DNA gyrase and topoisomerase OV mutations in the quinolone resistance-determining regions alter the clamp for fluoroquinolones. Dihydrofolate reductase variants bypass trimethoprim inhibition. Each edit is like filing down a keyhole so the key no longer fits. Efflux pumps act as bouncers. These protein complexes span membranes and expel diverse compounds, lowering intercellular concentrations below effective levels. Some are narrow and specific, others are broad spectrum, expelling multiple classes of antimicrobials. Overexpression can convert intermediate susceptibility into clinical resistance. Multi-drug efflux systems in gram-negative rods contribute to broad resilience against structurally unrelated agents, complicating therapy. Reduced permeability closes doors. In gram-negative bacteria, porins allow small hydrophilic molecules to cross the outer membrane. Loss or modification of porins reduces entry of Bt-lactams and other hydrophilic drugs. When combined with periplasmic Bt-lactamases, the effect is synergistic. Fewer molecules enter, and those that do are rapidly destroyed. Conceptually, it is like bricking up most windows and posting guards at the few that remain. Biofilms add a communal shield. Matrix slows diffusion. Metabolic heterogeneity dampens drug impact, and persister cells survive transient exposures. Resistance genes can be exchanged readily within dense communities, amplifying defenses. Biofilm-associated resistance is often phenotypic tolerance that reverses when cells disperse, yet clinically it mimics genetic resistance by reducing treatment efficacy. Horizontal gene transfer accelerates resistance spread. Plasmids carrying resistance cassettes move by conjugation across species boundaries. Transposons and integrons shuffle genes into new contexts, linking multiple resistance determinants together. Bacteriophages can mediate transduction of resistance elements. This genetic liquidity explains why a carbapenemase detected in one regional strain can appear across continents within years. Clinical implications are direct. ESBL-producing Enterobacterales may resist most penicillins and cephalosporins, leaving carbapenemes as the reliable bipalactam class. Carbapenemes E-producing organisms resist even those, forcing use of alternative classes often with more toxicity or less efficacy. Methicillin-resistant Staphylococcus aureus MRSA owes its name to an altered PBP with low affinity for nearly all bipalactams, a structural rewrite that blunts the entire tool class. Multi-drug-resistant Pseudomonas aeruginosa combines efflux, porin changes, and bipalactamases, creating layered defenses. Each example reinforces that resistance is not a vague label but a specific mechanism at work. A calm clinical vignette ties shield to consequence. A patient hospitalized with pneumonia shows initial improvement but then worsens on a beta-lactam. Cultures grow gram-negative rods carrying ESBLs. Without context, this looks like failure. With mechanism in mind, it is scissors at the gate, not inadequate dosing. Worsening shortness of breath, hypoxemia, or confusion in such a setting would be reasons to seek immediate professional care escalation. For you, the lesson is mechanistic clarity. Identify whether the shield is enzymatic destruction, target change, reduced entry, efflux, or communal biofilm protection. Reflection: when you learn to see resistance as scissors, filed locks, closed doors, or pumps, it stops being an overwhelming wall of acronyms. It becomes a set of stories you can follow, each with vulnerabilities at its seams. You turn the shield over and find an inscription: prevention, not repair, is strongest. The next step is stewardship, how disciplined use, de-escalation, and thoughtful combinations slow the forging of shields, conserving tools for when they are most needed. You stand before a desk stacked with ledgers. Stewardship is bookkeeping, accounting for which tools are used, how long, and when to close the ledger before waste invites loss. Antimicrobial stewardship is the disciplined management of these powerful tools to preserve their effectiveness, limit collateral harm, and slow the rise of resistance. It is not about withholding treatment, it is about precision, using enough for long enough, but not more. Start with initial choice. When a patient presents acutely ill, empiric therapy begins before cultures return. The empiric choice balances coverage of likely pathogens with risk of overtreatment. Broader agents are chosen when delay could be catastrophic: sepsis, meningitis, neutropenic fever. But once cultures and susceptibilities clarify the culprit, the stewardship principle is de-escalation, narrowing to the most targeted effective agent. De-escalation reduces collateral damage to the microbiome and decreases selection pressure for resistant bystanders. Think of it as trimming a floodlight into a focused beam once you see the target. Duration is the next ledger entry. Traditional courses were often prolonged out of caution, but evidence shows that for many infections, community-acquired pneumonia, urinary tract infections, cellulitis, shorter courses suffice once improvement is evident. Shortening duration without compromising cure reduces exposure, side effects, and resistance pressure. The concept is simple: stop paying rent when the building is already secure. Exceptions exist: deep-seated infections, prosthetic material, endocarditis, but the guiding principle is that treatment should be no longer than necessary. Combination therapy is another stewardship tool. Some combinations are chosen for synergy. A beta-lactam weakening the wall allows aminoglycosides to penetrate and act at ribosomes, producing enhanced killing against certain pathogens. Other combinations broaden empiric coverage initially but are pared down once results return. Combination therapy also prevents rapid resistance in special cases. Rifampin, for example, must be paired with other agents because single-step mutations render it useless alone. Stewardship ensures combinations are used for reasons, not by habit. Monitoring and allergy assessment matter. A frequent barrier to optimal choice is a reported allergy, often penicillin. Many such labels are historical and not true allergies. Some are childhood rashes unrelated to immunologic hypersensitivity. Stewardship includes clarifying allergy history and, when feasible, testing. Removing inaccurate labels reopens access to narrow, effective beta-lactams and reduces reliance on broader alternatives that foster resistance. Diagnostics are the steward's compass. Rapid identification tools, molecular panels, mass spectrometry shorten the time to narrow therapy. Biomarkers, such as procalcitonin trends, can support decisions to discontinue antibiotics when bacterial infection is unlikely. Stewardship programs integrate these data with clinical judgment, reducing unnecessary continuation when infection is not confirmed. Education and feedback keep the ledger balanced. Prescribers may overestimate risk of undertreatment and underestimate harms of overtreatment. Sharing data on resistance patterns, infection outcomes, and local antibiograms helps recalibrate those perceptions. Perspective audit and feedback, reviewing ongoing cases and suggesting adjustments, improve alignment with best practices without removing autonomy. The microbiome is a silent account. Broad-spectrum antibiotics deplete protective flora, allowing opportunists like Clostridioides difficile to bloom or resistant gram-negatives to colonize. Stewardship considers this collateral damage, sparing anaerobic flora when not required, avoiding repeated unnecessary courses, and recognizing that microbiome perturbation has long echoes. A brief clinical vignette demonstrates the ledger at work. A middle-aged patient hospitalized with fever and cough starts empiric broad-spectrum antibiotics. Cultures later yield a community-acquired respiratory pathogen susceptible to a narrow beta-lactam. Stewardship steps in, therapy is narrowed, duration shortened to evidence-based norms, and unnecessary second agents stopped. The patient recovers, collateral damage minimized. New rash with difficulty breathing, swelling, or wheezing during antibiotic administration in such a case would be reasons to seek immediate professional care to evaluate for true allergy. The teaching point is that accuracy and restraint can both protect the patient and preserve community tools. Reflection. Stewardship is less about saying no and more about asking how much, how long, and why this. With careful accounting, you extend the life of every tool, protecting not just this patient, but the next. You close the ledger and glance at a chart on the wall showing organ silhouettes: lungs, skin, urinary tract, gut, brain. Stewardship guides choices, but infections still announce themselves by organ systems. The next section maps which microbes are usual suspects in each site and why, giving you starting points before tests return. You unfold a city map at night and trace districts by their streetlights. Each organ system is a neighborhood with its own traffic rules, usual residents, and typical intruders. When symptoms arise, you start with place. Lungs breathe air and trap droplets. Skin touches surfaces and teeth. The urinary tract drains inward but opens to the world. The gut is a dense marketplace. The central nervous system, CNS, is a gated quarter behind tight barriers. Neighborhood first, suspects second, mechanisms always in view. Begin with the respiratory district. The upper streets, nasopharynx, oropharynx, sinuses, host resident flora and catch airborne particles. Viral introductions dominate colds and sore throats. Bacterial entries often follow after mucosal swelling blocks drainage. In acute bacterial sinusitis, think of common nasopharyngeal colonizers that ascend when ostia close. In acute otitis media, similar colonizers traverse the eustachian tube. For pharyngitis with tender nodes and sudden fever, a bet hemolytic coccus rises on the list. Cough and runny nose lower that probability. The lower streets, trachea, bronchi, alveoli, were designed to remain clean via cilia and cough. When microbes reach alveoli and multiply, the setting, season, and host narrow choices. Community-acquired pneumonia in otherwise healthy adults often points toward encapsulated respiratory cocci and select atypical agents that live inside cells and spread by droplets. Subacute cough with low fever and extrapulmonary clues leans toward the atypical group. In hospitals, intubation and recumbency invite water-loving gram-negative rods and a non-fermenting aerobes guild that thrives on equipment. Prior antimicrobial exposure shifts flora toward resistant lineages. Aspiration moves oral anaerobes and mixed flora into dependent lung segments, especially when consciousness is impaired. Red flags, respiratory distress, confusion, cyanosis demand urgent professional evaluation because gas exchange is the city's power grid. Skin and soft tissue behave like a perimeter wall. Intact keratin is dry, salty, and hostile to outsiders. Breaks invite residents and transients. Non-purulent cellulitis that spreads along tissue planes often implicates Bt hemolytic streptococci, which produce enzymes that ease passage through dermis. Purulent collections with focal tenderness and fluctuance fit coagulase-positive clusters that excel at abscess formation and toxin-mediated damage. Exposure refines the list. A puncture through rubber soles introduces water-associated organisms that favor moist materials. Animal bites mix mouth flora that include fastidious rods, adept at causing abrupt sepsis in certain hosts. Freshwater lacerations point to oxidase-positive aquatic rods. Saltwater injuries add helophiles that thrive in brine and can progress rapidly in those with liver disease. Chronic ulcers and neuropathic feet can harbor anaerobes and enteric rods under ischemic covers. Rapid pain out of proportion to findings, skin discoloration, crepitus, or systemic toxicity are cues to seek immediate care for possible deep fascial involvement. Here mechanism, enzymes that degrade fascia, gas-forming metabolism predicts velocity. Urinary tract infection, UTI, is the plumbing district. Sterile urine descends through ureters. The bladder sits behind a short urethra in many people, allowing ascent from perineal flora. Uncomplicated cystitis in healthy hosts typically stems from enteric rods that climb via adhesins designed to latch onto uroepithelium. New onset dysuria with frequency and urgency without vaginal discharge favors this pattern. Pyelonephritis when infection reaches the kidney adds flank pain and systemic signs. Catheters change architecture. Plastic surfaces acquire conditioning films and biofilms. Organisms with adhesins and urase enzymes, or those that excel at device colonization, predominate. Men with urinary obstruction develop stasis. Organisms reflect gut and skin access. In pregnancy, reduced ureteral peristalsis favors ascent, and even asymptomatic bacteriuria receives special attention in clinical practice because of maternal-fetal considerations. Red flags here are fever with back pain, vomiting, or confusion, signals for professional evaluation to assess for upper tract involvement or sepsis. Mechanistically, think ascent, adherence, and flow disruption. The gastrointestinal GI district is the crowded market. Most illness here arises from toxins or invasion. Abrupt, watery diarrhea after a shared meal suggests preformed toxins that disrupt ion transport without invasion. A longer incubation with fever and cramps implies organisms that attach and produce enterotoxins in situ. Dysentery, bloody diarrhea with fever indicates mucosal invasion or cytotoxin production by a short list of enteric pathogens. The tempo is sharper, the stool smaller in volume but painful. Long. A febrile diarrhea after travel can reflect protozoan adherence and malabsorption rather than bacterial invasion. Fatty stools and bloating with foul gas signal maldigestion. Antibiotic exposure reshapes the market profoundly. Spore-forming anaerobes bloom when competitors are suppressed, releasing enterotoxins that injure colonic epithelium. Seafood, eggs, unpasteurized dairy, and undercooked meats each cue specific suspects by reservoir, freshwater exposure, and camping hint toward protozoa. Daycare clusters suggest non-enveloped RNA viruses resilient on hands and toys. Severe dehydration, dizziness, minimal urine, persistent high fever, or blood and stool merit prompt professional care to evaluate severity and complications. Mechanisms: enterotoxin versus cytotoxin versus invasion, explain why some cases present with large volume watery stool and others with scant bloody mucus. The CNS is the gated quarter brain and spinal cord protected by the blood-brain barrier and cerebrospinal fluid CSF circulation. Bacterial meningitis arises when pathogens breach mucosa, enter blood, and cross into CSF. Which pathogen dominates depends on age, setting, and immune status. In adults, encapsulated respiratory pathogens remain prime suspects in certain communal settings or outbreaks. A fast-moving Diplococcus joins the top tier. In neonates and older adults, enteric gram-negative rods and specific group Becosi emerge due to perinatal and comorbidity patterns. In immunocompromised hosts, yeast with a thick capsule or molds with angio-invasive habits reach the CNS hematogenously. Subacute headache with raised opening pressure points toward encapsulated yeasts, whereas focal neurologic deficits and ring-enhancing lesions in severe T-cell deficits expand the list to protozoa. Encephalitis, parenchymal inflammation, leans viral with neurotropic enveloped DNA viruses establishing latency and reactivating, and arboviruses arriving by mosquitoes. The classic triad of fever, neck stiffness, and altered mental status in meningitis is variably present, but any combination of severe headache, photophobia, confusion, new seizure, or focal neurologic signs warrants immediate professional evaluation. Mechanistically, capsules evade splenic filtering, endothelial interactions permit crossing, and host cytokine storms swell confined spaces. A few cross-district principles help you move calmly. First, time course. Hyperacute hours suggest toxins, fulminant bacteria, or vascular invasion. Subacute days suggest intracellular pathogens, dimorphs, or early immune-mediated patterns. Indolent weeks suggest mycobacteria, some fungi, or parasites with cyst stages. Second, host factors. Asplenia raises risk for encapsulated bacteria. Neutropenia opens doors to molds. Complement deficiencies favor specific diplococci. Structural lung disease selects for non-fermenters. Travel and exposures redraw the map. Third, devices change everything. Endotracheal tubes, central lines, prosthetic joints, and urinary catheters replace mucosa with plastic, inviting biofilms and introducing water-associated organisms. Fourth, anatomic shortcuts. Sinus orbit brain contiguity, ear mastoid dural venous channels, dental deep neck spaces. Spread respects planes but ignores your naming conventions. Illustrate with 3 brief cameos. A community-dwelling adult develops fever, productive cough, and pleuritic pain in winter. Focal crackles on exam and lobar opacity on imaging favor an encapsulated respiratory coccus acquired in the community. A teenager with a painful fluctuant skin lesion after contact sports carries a suspect adept at abscess formation and toxin production. Clustering on culture points the way. An older adult with new dysuria and urgency after starting a catheter has a device-associated UTI. Biofilm builders and urase producers rise on the list. In each, worsening systemic signs, hypotension, confusion, or rapidly spreading pain are cues for urgent professional care. A quiet reflection. When you ask first which neighborhood, and only then which usual suspects live there, you replace scattered lists with a street map. Place, tempo, host, and device tell a coherent story before any report arrives. You fold the map and glance at the hospital's doors and corridors. Suspects ride on hands, droplets, and surfaces, as much as in blood. Barriers, isolation, and cleaning break chains at their weakest link. How do you place a mask, glove a hand, or choose a disinfectant to keep one neighborhood's fire from jumping the alley to the next? You walk the halls of a hospital at dawn. Doors open, carts roll, and countless hands touch bed rails, keyboards, and tubing. Infection control is this choreography, a set of movements that block invisible passengers from crossing thresholds. The principles are not mystical, they are simple physics and biology. Droplets fall, hands transfer, spores persist, air currents drift. By anticipating routes, you interrupt chains. Start with isolation, the placement of patients by risk of transmission. Standard precautions apply to all: hand hygiene before and after contact, gloves when touching blood or body fluids, masks when splashes are possible. Transmission-based precautions layer on top. Contact precautions, gowns and gloves prevent spread of organisms shed heavily into the environment or persisting on skin, such as multi-drug-resistant enteric rods or spore-forming anaerobes. Droplet precautions, surgical masks within a defined radius, interrupt organisms propelled in respiratory droplets that fall quickly, such as respiratory cocci and influenza viruses. Airborne precautions, respirators and negative pressure rooms protect against smaller particles that remain suspended, such as Mycobacterium tuberculosis or varicella virus. The logic follows particle physics: heavier droplets drop, fine aerosols float, spores settle but remain viable for months. Personal protective equipment, PPE, forms the visible barrier. Gloves protect hands but must be removed without contaminating skin. Gowns prevent contamination of clothing and transmission to other patients. Masks shield mucous membranes. Respirators add filtration of aerosols. Eye protection is needed for splashes. The barrier is only as strong as the donning and doffing technique. A misstep in removal can undo the protection. The rule is clean to dirty, inside to outside, discarding each layer as you peel. Hand hygiene is the quiet constant. Alcohol-based hand rubs denature proteins and rapidly inactivate many organisms, but spores resist and require soap and water. Contact time matters. Rubbing until hands are dry ensures effectiveness. Gloves do not replace hand hygiene. They add a barrier but can transmit between tasks if not changed. Visualize every touch as a possible transfer of organisms between microenvironments. Sterilization and disinfection manage objects. Sterilization achieves complete elimination of all microbial life, including spores, achieved by methods such as steam under pressure, autoclaving, dry heat, or chemical gas. Disinfection reduces harmful organisms but may spare spores or some resilient viruses. High-level disinfectants inactivate most pathogens, including enveloped and non-enveloped viruses and fungi. Intermediate-level agents may fail against spores and some non-enveloped viruses. Low-level disinfectants reduce vegetative bacteria and enveloped viruses, but not hardier forms. Cleaning precedes disinfection. Organic matter shields microbes from chemicals. Choosing the right level depends on object use. Surgical instruments must be sterilized. Endoscopes require high-level disinfection. Bed rails may suffice with low-level but frequent cleaning. Environmental reservoirs require vigilance. Water systems can harbor biofilms with aquatic gram-negative rods that aerosolize in showers or humidifiers. Air systems must filter and maintain pressures. Negative pressure keeps airborne pathogens from leaving rooms. Positive pressure protects immunocompromised patients from outside microbes. Surfaces accumulate spores and resistant organisms. Frequency of cleaning, choice of disinfectant, and staff adherence determine whether those surfaces act as sinks or barriers. Outbreaks test the choreography. A cluster of similar infections in a ward may reflect a single contaminated device, a colonized staff member, or breakdown in hand hygiene. Epidemiology teams track cases, map exposures, and sometimes perform molecular typing to link strains. Control measures include cohorting patients, restricting admissions, deep cleaning environments, and reinforcing PPE use. The logic is containment. Stop new cases while extinguishing existing reservoirs. Device management is central. Central venous catheters, urinary catheters, endotracheal tubes each breaches barriers. Bundles of best practices reduce infection risk. Strict asepsis at insertion, daily review of necessity, meticulous maintenance, prompt removal when no longer required. Each day a device remains, risk rises. The mechanism is clear. Foreign material lacks immune defenses, allows biofilms to form, and becomes a scaffold for microbial persistence. Vaccination plays a less visible but crucial role in infection control. By reducing carriage and transmission of specific pathogens, vaccines protect both individuals and communities. For instance, conjugate vaccines against encapsulated respiratory bacteria reduce invasive disease and decrease asymptomatic carriage, indirectly shielding those who remain vulnerable. A focused clinical cameo. An inpatient with severe diarrhea tests positive for a spore-forming anaerobe after broad antibiotic exposure. The organism sheds spores that resist alcohol-based hand rubs and linger on surfaces. Contact precautions with gowns and gloves, soap and water hand washing, and dedicated equipment help contain spread. If new fever, abdominal distension, or confusion develop, those would be cues for immediate professional evaluation to assess for complications such as toxic megacolon. The lesson is precise. Root plus persistence dictates barrier choice. Reflection: when you treat control not as punishment but as choreography, placing masks for droplets, respirators for aerosols, soap for spores, you see order instead of burden. The invisible passengers move by rules, and rules can be broken with discipline. You leave the ward and enter a lecture hall with chalkboards showing numbers, basic reproduction ratios, transmission chains, epidemic curves. Infection control at the bedside widens into epidemiology at the population level. How many secondary cases arise, how surveillance spots clusters, and how interventions bend curves. You step into a hall where chalkboards stretch along the walls, filled with branching lines and numbers. Epidemiology is the study of how infections move through groups, tracing not a single patient but chains of transmission across a population. The same principles that describe a spark in dry grass describe microbes spreading in communities. The math is shorthand for the biology you have seen. Begin with the basic reproduction number, R0. It represents the average number of secondary infections caused by one infected individual in a fully susceptible population. If R0 is less than 1, each case produces fewer than one new case on average, and transmission dwindles. If R0 is greater than 1, the chain tends to grow, and outbreaks can occur. R0 is not a fixed property of the pathogen alone. It reflects infectiousness, duration of contagiousness, and contact rates. A virus with a high R0 spreads easily in close quarters. A bacterium with modest transmissibility requires particular conditions or vectors. Conceptually, R0 is the slope of the first line in the chain. Above 1, the line ascends, below 1, it falls. Chains of transmission form the backbone. Each link is an exposure: respiratory droplets inhaled, food contaminated, vectors biting, devices breached. Direct transmission moves from host to host without intermediates, cough to mucosa, hand to wound. Indirect transmission involves vehicles such as food, water, or fumites, or vectors such as mosquitoes. Some chains are short and explosive, point source outbreaks when many are exposed at once to a contaminated meal. Others are propagated, one case seeds the next, leading to epidemic curves that rise, peak, and fall. Herd immunity is a population-level shield. When enough individuals are immune through infection or vaccination, the average number of susceptible contacts per case falls. Even if a pathogen has a high R0, immunity can reduce the effective reproduction number R below 1, halting spread. The threshold fraction immune needed for herd protection is roughly 1:1 AR0. For a highly transmissible agent with R0 of 10, 90% must be immune to block chains. For one with R0 of 2, 50% suffices. Vaccines that reduce carriage as well as disease amplify herd effects, lowering circulation even further. Surveillance tracks these movements. Passive surveillance collects routine reports of cases. Active surveillance seeks cases deliberately, contacting providers or testing populations. Syndromic surveillance uses patterns of symptoms, such as sudden rises in respiratory complaints, to detect outbreaks even before specific diagnoses are confirmed. Laboratory networks contribute by identifying unusual strains, resistance genes, or new clusters. Together, surveillance systems create early warning and inform interventions. Epidemic curves tell stories in graphs. A sharp spike followed by decline suggests a common point source, such as contaminated food at a single event. A gradual rise and fall with successive peaks suggests propagated spread from person to person. A curve with multiple peaks spaced apart suggests intermittent exposures, such as repeated contamination of a water source. Reading curves helps epidemiologists identify the type of outbreak, estimate incubation periods, and time interventions. Contact tracing is an application of chain logic. When an infectious case is identified, tracing identifies close contacts during the contagious window, tests or treats them as needed, and interrupts further spread. This approach has been crucial for diseases with defined incubation periods and identifiable transmission routes, such as tuberculosis, meningococcal meningitis, and emerging viral infections. The goal is to snip links before they forge new branches. Vector ecology adds another dimension. Mosquito-borne diseases depend on vector density, biting behavior, and environmental conditions. Control measures, bed nets, insecticides, environmental modification target vectors rather than pathogens directly. Seasonality in many infections reflects vector cycles or environmental survival. Winter peaks for respiratory viruses in temperate climates, rainy season peaks for mosquito-borne diseases in tropical regions. Outbreak investigation follows a structured path. First, establish that an outbreak exists by comparing case numbers to expected baseline. Next, define a case with specific clinical and laboratory criteria. Then describe cases by time, place, and person, epidemiologic triad to identify patterns. Generate hypotheses about sources and modes of spread, test them with analytic studies, and implement control measures. The loop closes with communication and prevention planning. Each step is systematic, reducing chaos to method. A brief clinical vignette makes this tangible. In a college dormitory, several students develop sudden fever, headache, and stiff neck over 2 days. Rapid recognition links cases. Investigation shows a shared serogroup of an encapsulated diplococcus. Prophylaxis for close contacts and vaccination campaigns halt further spread. If a patient with this syndrome develops a purpuric rash, shock, or altered consciousness, those are urgent cues for immediate care. The epidemiologic lesson is that clustering in time and place, plus severe presentation, signals a chain that must be broken fast. Reflection: when you see cases as points on a map connected by lines, the story expands from individual to community. Numbers are not abstractions, they are translations of who infected whom, and whether tomorrow's line is longer or shorter. You close the chalkboards and walk into a side room with case files stacked neatly. Numbers and chains become stories again. Guided cases where initial presentation leads through reasoning to likely culprits, testing, and conceptual management. You lay 3 slim folders on a quiet desk: sore throat, burning urination, red-hot skin. Guided cases are not recipes. They are rehearsals that turn presentation into mechanism into likely actors, and actors into focused tests and concept-level first moves. You will read each folder the same way: place, tempo, host, exposures, red flags, then let biology trim the list. Folder 1. Acute pharyngitis. The room is winter dry. A patient reports sudden throat pain, fever, tender anterior neck nodes, and no cough. You start with place, an oropharynx lined by lymphoid tissue, bathed in secretions and exposed to droplets. Tempo, abrupt. Host, otherwise healthy. Exclusions matter: cough, rhinorrhea, hoarseness, and conjunctivitis. Tilt you toward viral etiologies, adenoviruses, rhinoviruses, other respiratory RNA viruses, because they inflame multiple upper airway sites simultaneously. Absence of those features with high fever and anterior cervical lymphadenitis increases the probability of a beta hemolytic coccus that specializes in pharyngeal adhesion and immune mimicry. Mechanism makes this plausible. Streptococcus pyogenes binds epithelium via lipoteichoic acid and M protein, secretes enzymes that breach superficial defenses, and provokes a brisk neutrophilic response that makes swallowing exquisite. A fine sandpapery rash or a strawberry tongue signals toxin-mediated scarlet features. Palatal petechiae and tonsillar exudates are supportive but not diagnostic alone. You translate probability into tests. Rapid antigen detection assays capture streptococcal cell wall antigens within minutes. Sensitivity varies with technique and organism load. A negative rapid test in a child or adolescent with high clinical likelihood may be followed by throat culture, which remains the reference for specificity and picks up low-load infections. In adults with lower pretest probability, a negative rapid test often suffices to end the search. You resist overinterpreting a superficial swab that shows a carrier state. Colonization without tissue invasion is common. Red flags redirect the script. Muffled hot potato voice, drooling, trismus or unilateral peritonsillar swelling implies a deep space process. Severe neck pain and swelling with systemic toxicity raises concern for suppurative thrombophlebitis of a jugular vein. New dyspnea or stridor signals airway risk. Those constellations merit immediate professional evaluation because anatomic compartments are tight and inflammation spreads along fascial planes quickly. Your conceptual first moves, purely educational, follow mechanism. When a true streptococcal infection is confirmed, narrow, targeted therapy is typically considered in practice to reduce complications and shorten symptom duration, while avoidance of antimicrobials in likely viral presentations preserves the microbiome and curbs resistance pressure. Supportive care aligns with pathophysiology, hydration for mucosal comfort, antipyretic strategies for cytokine-driven fever. The takeaway for you is not a prescription but a map. Symptom sieve causes, a rapid antigen test sharpens, deep space signs escalate. Folder 2. Uncomplicated lower urinary tract syndrome. A healthy, non-pregnant adult notes dysuria, frequency, urgency, and suprapubic pressure without flank pain or fever. Place the bladder's urothelium. Tempo, hours to a couple of days. Path, ascent from perineal flora along a short urethra with uroepithelial adhesins as grappling hooks. Mechanism centers on uropathogenic Escherichia coli using P and type 1 fimbriae to bind mannicillated receptors, resisting washout. The bladder responds with cytokine-driven urgency and local inflammation that makes voiding burn. Other enteric rods and a few gram-positive cocci can play supporting roles. Sexually transmitted pathogens are less likely with classic irritative voiding without discharge or dyspareunia, but enter the differential when exposure history shifts. Testing reads the chemistry of inflammation and the footprints of gram-negatives. A clean catch urinalysis offers leukocyte esterase, an enzyme proxy for neutrophils, and nitrites produced when enteric bacteria reduce dietary nitrates. Remember that not all uropathogens reduce nitrates, so a negative nitrite does not exclude infection. Microscopy shows pyuria and sometimes bacteria. Culture quantifies organisms and identifies species, guiding susceptibility when needed. In simple first episode syndromes with classic features, many clinicians proceed without culture in practice, but cultures gain weight with recurrence, atypical features or risk factors. Distinguish carefully between contamination, skin or vaginal flora in the sample, colonization, asymptomatic bacteria, and infection, symptoms with compatible findings. Asymptomatic bacteria is its own category. In most non-pregnant hosts it is not treated in practice, whereas specific contexts, pregnancy, certain urologic procedures shift that calculus. Red flags reframe urgency. Fever, flank pain, nausea, vomiting, or systemic symptoms suggest pyelonephritis, renal parenchymal involvement with ascending infection or bacteremia risk. Catheterization rewrites the script. Devices invite biofilms, change species prevalence and transform interpretation. New systemic signs or obstruction concerns merit prompt professional care. Concept level first moves emphasize hydration to support flow, and when bacterial infection is likely, targeted agents chosen with awareness of local resistance patterns and individual factors. Stewardship trims duration in many straightforward cases. If severe pain, high fever, or confusion develops, the map switches lanes toward evaluation for upper tract involvement or sepsis. Folder 3. Cellulitis and abscess. A patient presents with a warm, expanding area of erythema on the calf after a minor scrape. Place dermis and subcutaneous tissue. Tempo day by day. Non-purulent cellulitis with diffuse, ill-defined borders and lymphangitic streaking leans toward bakemolytic streptococci that secrete enzymes to traverse tissue planes. Purulent, focal, fluctuant lesions point toward coagulase-positive clusters that excel at abscess formation. Toxins and leukocytins carve pockets where neutrophils and bacteria struggle in low oxygen, acidic microdomains. Ultrasound at the bedside can reveal a fluid collection invisible to inspection, changing management because a pus-filled cavity often requires procedural drainage to resolve. This is mechanism, not dogma. Fluids under pressure block perfusion and buy protection for bacteria that wall agents alone may reach poorly. Exposure reshapes suspects. A puncture through a rubber-soled shoe introduces water-associated gram-negatives, adept at moist materials. Dog or cat bites add fastidious or encapsulated mouth flora. Freshwater or brackish water injuries Add oxidase-positive aquatic rods, and seawater adds halophiles that move fast in those with chronic liver disease. Chronic edema from venous or lymphatic compromise predisposes to recurrent streptococcal cellulitis by lowering skin defenses. Tinea pedis maceration opens interdigital portals. Red flags demand speed, severe rapidly progressive pain out of proportion to findings, bullae, skin anesthesia, crepitus, violaceous discoloration or systemic toxicity signal possible necrotizing soft tissue infection, where bacterial enzymes and toxins outrun host defenses along fascial planes. Those findings are cues for immediate professional evaluation because hours matter. Mechanism explains velocity, synergy of anaerobes and aerobes lowering redox potential, toxin-mediated vascular thrombosis, starving tissue ahead of the visual edge. Your conceptual first moves match biology. For non-purulent cellulitis in immunocompetent hosts, streptococci dominate the early differential. For purulent infections, coverage for coagulase-positive clusters becomes central in practice, with procedure decisions driven by the presence of a collection. Cultures from intact skin rarely help. Aspirates or drainage specimens, when obtained, carry more meaning. Imaging is selective, ultrasound for abscess, advanced imaging if deep space spread or osteomyelitis is suspected by exam or persistent course. Device or prosthetic involvement reclassifies risk because biofilms change susceptibility patterns and clearance. Across the three folders, patterns repeat. Place narrows suspects, tempo distinguishes toxins from invasion, host factors, pregnancy asplenia, neutropenia, diabetes, tilt the board, devices and exposures redraw it. Tests work best when matched to biology. Rapid antigens in a clean oropharyngeal presentation, urinalysis where nitrite physiology applies, ultrasound when pus changes the plan. Red flags are not add-ons, they are the points where anatomy and mechanism raise stakes. Airway, kidney, fascia, bloodstream. A brief reflection. Practicing the same disciplined reading over different complaints teaches your hands to move calmly, choose what to test, what to ignore, and when to escalate, while remembering that education is not prescription and thresholds for seeking care are part of the map. You slide the folders back into their sleeves and look toward a final quiet table. All the streets, tools, and signals you've gathered can fit on a single page. Pathogen, organ approach, and a slow exhale to close the night. Which landmarks make that one page trustworthy enough to rest on? You sit at the quiet table and smooth out a single page. It is not filled with paragraphs, only a map, arrows between microbes, organs, and conceptual tools. This is not simplification for its own sake. It is condensation, reducing complexity to landmarks that orient you when fatigue clouds detail. Begin with the architecture of the map. Along one axis, list organs and systems: lung, skin, urinary tract, gastrointestinal tract, central nervous system. Along the other, place common pathogen families: cocci, rods, viruses, fungi, parasites. Arrows connect where biology and exposure align. The map is not exhaustive, but it reminds you of the most likely partnerships. In the lung, encapsulated respiratory bacteria lead in community settings. Atypical intercellular agents add when cough and extrapulmonary clues align. Viruses dominate in seasonal waves. Molds and yeasts emerge in immunocompromised hosts. In the skin, bet hemolytic streptococci spread along tissue planes. Coagulase-positive clusters form abscesses. Animal and water exposures add fastidious rods. Chronic edema and breaks in keratin change risk. In the urinary tract, ascending enteric rods dominate. Catheters add biofilm builders. Pregnancy alters physiology and heightens stakes. In the gastrointestinal tract, toxin producers cause abrupt watery losses. Invasive enteric bacteria and parasites cause bloody stool. Protozoa cause chronic malabsorption. Antibiotics predispose to spore-forming anaerobes. In the CNS, encapsulated bacteria breach barriers. Specific viruses invade neurons. Yeasts with capsules and molds with vascular tropism target the immunosuppressed. Parasites form cysts. Against each pathogen family stand classes of tools. Wall-active agents halt cocci and rods, where peptidoglycan is essential. Ribosome blockers slow protein assembly in both gram-positives and gram-negatives, with special reach into intracellular niches. DNA gyrase and RNA polymerase inhibitors halt replication and transcription. Folate pathway inhibitors strike organisms that must synthesize what you consume. Antifungals pry ergosterile membranes, saw glucan walls, or smuggle in false nucleotides. Antiparasitics disrupt anaerobic DNA, microtubules, or neuromuscular conduction unique to worms. Each class fits a place, not by habit, but by mechanism. Resistance landmarks stand beside them. Enzymes hydrolyze beta-lactams, altered PBPs blunt binding, methylated ribosomes repel macrolides, mutated gyrase resists quinolones, efflux pumps and porin loss starve drugs of access, biofilms shelter persisters. Stewardship keeps pressure lower by narrowing empiric choices, shortening durations, and discarding unnecessary double coverage. Inaccurate allergy labels are erased to restore access to narrow tools. Diagnostics Guide, Gram stain, antigen tests, NATs, serology, each with timing caveats, each clarifying who is present and when. The page also holds red flag cues to remember. In respiratory illness, severe dyspnea, confusion, cyanosis. In skin infection, pain out of proportion, necrosis, bullseye. In urinary infection, fever, flank pain, confusion. In gastrointestinal illness, severe dehydration, blood in stool, abdominal rigidity. In CNS disease, altered consciousness, new seizure, purpura. Each is a signpost to immediate professional evaluation. Reflection. The map's power is not detail but proportion. You no longer memorize every organism. You recall patterns, connect mechanisms, and notice when a case diverges sharply from the usual streetlights. That divergence is itself a guide. You fold the page and let your breath slow. Threads from microbiology, pharmacology, diagnostics, resistance, stewardship, and clinical vignettes weave into one cloth. You recognize that each fact was not separate, but part of a single system. Microbes adapted to niches, humans responding with defenses, tools carved at weak points, and communities shaping transmission. The night has been long, the corridors many, the cases varied. You have traced organisms from invisible walls to epidemic curves, watched tools fit locks, and seen shields rise. What remains is not the clutter of lists, but the calm of structure. Place, mechanism, and pattern will guide you when detail fades.